No, I think at this point we go up to 15 and
Get to my starting point.
get to the starting point. Yeah, just north of that wall.
So you're heading towards starting point of the patch test.
How are we naming this starting waypoint? So the starting waypoints. Oh,
Yes?
we're naming these lines using the starting waypoint.
Okay. Which I can kind of see from here, but yeah. Okay.
That's different from what we did last time.
Yes,
Which was different from what we did the time before. That's great. Yes.
I would just like it known that I changed which thing was visible on each screen, but I did not change that map thing. I don't know how that happened.
What did you do?
I didn't do anything.
We give you the iPad for 1 minute.
I didn't look like you. I didn't do anything. I didn't even do anything on that screen.
I know how to fix this.
It's just
I think I know what
essentially
you want, but
1 meter was.
This is what happens when you let Charles.
Yeah, well,
I saw that in real time happening last night. It was mostly right. There's just a few zero values tossed in there. But you couldn't actually see hard.
Well, hello and welcome to Divestream 611 of the remotely operated vehicle Sebastian. We've just touched down on an active hydrothermal site known as Iguanas West, part of the Iguanas hydrothermal vent field. We're just scoping the bottom here briefly, and then we're actually going to hop back up a little ways off the bottom, about 15 meters altitude. We are going to begin this dive stream, with a mapping and patch test using the InSAS, which is our synthetic aperture sonar. Estimating our dive length at about 9 hours or so. So not super long. We've actually got to go pick up the hydrophones this evening. But we're just getting started with the dive stream portion here. So again, we're looking to use the InSAS for the first few hours, along with some magnetometer surveying. And then after that, we'll be dropping down for the last few hours to do some visual survey and sampling. We are expecting active venting at the imaging and sampling sites. Going to be potentially collecting some bamboo corals, also keeping our eyes peeled for any skate eggs. We might collect some of those. Rock samples, of course, are usually a given for this sort of thing. Looking to take some water samples with the Niskins on board ROV Sebastian. And if we find some nice sediment near active vents, we are going to hopefully take some push cores for sampling. Time permitting, we'd love to take some photomosaics as well, and potentially some glass sponge samples. Other than that, just kind of continuing our exploration. So again, welcome, 611. Just catching us at the beginning, but we will be hopping up off the bottom here shortly. We're going to be staying around this altitude, actually, about 15 meters. So we can just barely make it at the bottom, but we are going to be up. And unfortunately, there is no waterfall display or anything like that for the InSAS, so it'll mostly just be kind of shadowy glimpses at the bottom. and the voices of the folks in mission control. Across the front, we'll have John. In the pilot seat, we have J-Rod. Co-pilot is Coco. And then I'm currently in the seat over here, Alex, multimedia technician. But lunch is coming up in just a couple minutes, so people will be shuffling in and out before we kind of settle on our afternoon. So welcome to anyone just joining us.
Alright, that's one turn, going back the other way.
Monica, what did you do?
This is stressful.
Should I try zooming out?
I got it.
Alright, I'm gonna start going.
Okay. So this is start with line 1.
Oh, that's a dangerous thing to do.
Let me get going.
0, 0, 1.
And we're gonna go that one. That's 0.3. I'm going just, I got it set for 0.2 right now.
What's that? Well, we saw the cliff, so we know where north of it which was the key. There we go.
Alright, I'm coming up to the actual start point. Seems to be stable. I'll fix it.
Start line
Yeah.
0, 0, 1. Yeah, Maurice was going to come down and someone
Ooh, there's a question in the chat already.
Yeah, I'm kind of liking this one. I think it's about the use of InSAR, synthetic aperture radar, and how that might compare to InSAS, which is our sonar.
So, let's see, the question is the InSAS the same similar to what astronomers do by taking pictures of the same target 6 months apart or by using multiple telescopes all across the globe?
Oh, I guess they're looking
They're talking about-
upwards. Yeah,
We're
because
talking
they
about stacking.
they're using stacking. Okay,
Yeah.
because there's InSAR, which is used from or was used from the space station and the satellites to look downward. But I guess this question's looking upward.
Yeah, so, but the principle is I can see why they're asking this question. It's actually a pretty good question, but it's not really the same principle. It is in that you're getting a better resolution image by doing things multiple times. But with the astronomy one, it's a case of stacking over and over and over.
Right. Yeah.
Whereas what we're doing, which is, as you said, very similar to synthetic aperture radar, which is what satellites do when looking down at Earth, is hitting the same object from essentially different angles, different positions.
Yeah.
So our platform of observation is moving and seeing the same target from multiple angles. Yeah, that's a good question.
Yeah, because with the InSAS that's on Sebastian, there's like receivers on each side of the sub. Is there one transmitter on each side?
Yes,
But then there's multiple receivers.
exactly.
Yeah, so they're getting just slight variations. Yeah.
Exactly.
How many are there per each side of receivers?
One array is 16, but two stacks of 16, so 32 on each side.
32 on each side.
Yeah.
Okay.
Our pitch is remarkably stable. Oh, I should have. Sorry. I'm not looking at that number at all.
Yeah, we changed where we have the lift point at, so it should be better, but still have
Move
to
it
get everything
forward or
forward.
yeah. We could just see the seafloor.
So like I said, we're going to have lunch starting up here. So you'll hear folks kind of shuffling in and out.
Anyway. No, Charles overlaid it over the ship ultimately, and he said no shipping required. So I'm guessing what we collected, they probably did that. So this is fun.
Yeah, nobody wants to put up their flags.
Yeah, you drape them.
Yeah.
That's okay, I think they're close enough. Right. And which waypoints are those? 611. So there were 3 CSV files. Did those get loaded?
Sonar.
Not 100% sure because I wasn't here,
Yeah.
but
Let me have a minute and do this.
so we want these with results there, right? These are different.
We don't need these ones on flare mouse.
Okay.
We speed up just a touch.
Let me think about
I
this.
would bridge speed up just the smallest bump.
What it would be good is to have a weekly,
Yeah, go ahead, Arabi.
files 2 and 3 on the mouse.
Yeah, no problems at all. Point 7, that'd be good.
And this is file 1.
This is file 1. I don't know if 2 and 3.
Point 6, yeah, no problems. I'll do that
This
next.
is not in CSV.
Okay. Yeah, we'll have to ask.
I'll need something for me.
Yeah, are you aware of the waypoints? There's 3 CSV files.
Yes. We just have the one loaded for now so it doesn't get chaotic.
Okay. Okay, then we're over.
Okay. Awesome.
Thank you.
Sure.
John, we've got a question of relation of where we are. So they want to know if the Iguanas Pinguinos vent field is located to the west of Eye of Mordor Seamount.
That is absolutely correct. We are, how far west are we? Let's see if I can figure that out. Bear with me, everybody.
No worries.
Yeah, we're about 100 kilometers or so west of Eye of Mordor.
Okay.
I bet they asked that question so we could say Eye of Mordor over and over again.
Yeah.
That's the only reason we're going there.
Do you have a range to drop in when we get there?
So
Or?
we have a. I'm sure someone has a ring they don't need anymore. There is a black smoker at a vent field called Lokachev on the Mid-Atlantic Ridge. That's called Badadur. And it is very tall, very skinny, and it has absolute belching black smoke coming out the top. So there's Lord of the Rings references littered all over the seafloor.
Yeah, for folks just joining us, we are up about 15 meters off the bottom here at Iguanas West. We are going to remain 15 meters off the bottom for the first little portion of this dive. We're going to cruise around for a few hours with our synthetic aperture sonar, as well as the magnetometer. So in order for that, we're up off the bottom. After that, then later in the dive, towards the end, we're going to drop down and actually do imaging and sampling of what's expected to be pretty active vents, I think. So. Our estimated dive length is 9 hours, so shorter than our more recent dives, but longer than the last dive, which was just picking up those current meters.
This is what we call type 2 fun.
Type 2 fun, yeah.
The gratification comes later when we have the data.
Yeah.
It's like hiking up a mountain.
I was going to say, there's people that consider hiking like a type 2,
Yeah.
where the actual process, but the destination.
Sure feels good when you get there. But then part 2 of this dive will be type 1 fun.
Yeah, yeah.
I always put a 0 day whenever I go backpacking, at least 1 0 day. That makes it even better.
Oh, just a lazy camp day?
Yeah.
Yeah. Don't move sites, or if you do, I move it like 45 feet because you have to because of the move.
Yeah, yeah.
Alex, the sit has probably a better image for than now.
Kind of pointing down. Yeah.
You can see before. I can try to make that better here a little bit.
No worries. Weather isn't looking too bad today. So we'll keep an eye on it, but looking pretty good for the stream.
Do you want to overshoot the line a little bit?
Yeah. Yes, by 10 meters.
I will do my best once I get there.
Do you want to grab some lunch, Hope?
Sharice was gonna come back down.
Okay.
And then I assumed
All right, it's just changed.
yeah.
So how wide out a swath are we getting then at 15 meters?
It's 10 times altitude, so it's about 150 meters.
On each side? Okay. So yeah, the InSAS is reaching out about 150 meters on each side right now. And
You can stop whenever you want.
Right, just overshooting. So we can do the same thing on the way back.
how to keep your speed.
What's that?
Is that like going through the finish line with a constant speed? Is that what that is?
And then also to be able to give me a run up coming back the other way. Going a little bit faster at the end, but it seemed to stay pretty stable.
Okay. End of line.
End of line 001.
Yes. Thank you.
I have a question on differences between day and night dives, if there really is a noticeable one as far as what we're seeing. Is there a circadian rhythm still? And on the way down, the water column can certainly change depending on when we're diving, because all the deeper critters can be making their way up in the evening to be up in shallow water. So if they're migrating actively, that can be a little more activity. And then I know that venting sites have been shown to be impacted by tides, but probably not so much whether it's night or day, just tides.
There have been measurable changes in temperature of vents, but as you said, that's probably a pressure effect from the tides, the water over the top. But yeah, day night, not that I know of.
Not down here.
Starting my move.
So last night, before we headed over here, we did drop off the hydrophone lander again. And that was currently set to about a 24-hour deployment. So after this dive, we'll be going to scoop that lander back up, see what sounds it may have recorded.
We are looking into the possibility of going to collect it without having to recover the ROV.
Oh, just kind of slide on over there.
Well, I didn't think we could do that. So it's about 1 kilometer away from us
Okay.
now. So I thought that was a non-starter in terms of keeping the ROV on bottom. But the other option is with the
Pinging. We can start pinging. Yep.
Yeah, depending on what time.
weather and time and all
Yeah.
that stuff. But if we could do that, then this dive,
Would extend.
we wouldn't have to end it at whatever, 3, 4, 5 o'clock.
Right, because the idea was that we would recover, go get the lander, and then go back in.
Back again.
Yeah.
So if we could avoid that.
Yeah. We'll see.
We said we're a kilometer away. Is that along access?
Mostly along axis. Let me see here. The lander drifted 150 meters north of the axis. Pretty close, yeah.
All right.
Yeah. It was 20 something gigabytes. 20 gigabytes. One range. 17 minutes. So 1 gig
Gigabytes a
a
data in
minute.
17 minutes.
So approximately 1 gig a minute.
1 gig a minute.
It's like our 4K camera.
That's why most of my luggage consists of hard drives.
Guys seen anything unusual? Any skates? Eggs? Anything just casually pop into your mind?
Pillows.
Is that?
Pillows.
Pillows.
Everywhere.
That might be a little bit of sediment right there.
Yeah.
The nice thing about the SAS is you can just see the seafloor.
And the bad thing about the SAS is that you could just
You can
see
just- This is how we filter out the biology. We keep the geology. We can see all the features, but can't see any of the animals.
Well played, sir.
We found our ideal survey height.
And we can map at the same time. Oh, she knows.
Yeah, it looks to me like you picked a really good spot.
Yeah, it's great. And I think the depth only changed like 5 meters across 300 meters, so that's really nice.
What was our threshold? 10 meters?
A bit more, I think. We did way more earlier.
Doing SAS right now. Trying to keep the distance slightly ahead of us. On the same speed. Currently 0.6 and then. Okay. Yes. 0.3. But I started them off at a 0.5 and then I was slowly catching up to 0.6. That worked. I'm just monitoring this line to make sure I'm not going to have to bring him back to 0.5 if he goes too much. I'm just watching the local.
Can we get that some sort of a ground effect for them so that they can monitor it?
They have it.
Okay, they can compare to
He has the same screen on that. But it's just double or whatever. If we want to go 0.3, 0.6.
Oh, okay.
Wait for the next shift.
Yeah, so once I get to the table or
Your feet are
you're the lodging much more.
Yeah, who's
You
that?
asked me on Teams to continue logging.
Just so you can get.
I'm logging right now. I sent Hope away for lunch.
Because you just need a little bit of a run up. Like so when y'all go to do this one, you're actually gonna have to set up
What's
over
that?
here. Yeah. And then get a run
Are you
up.
asking what's happening, or is that
Go.
be what's happening?
So I started them first. They get moving.
Uh,
I went to see the
SAS map.
Something.
That's the old one. Then I start going towards the.
Yes, it is.
And
The new bridge, so
start
it has
slowly
ability
speeding up to a 0.3.
but I have trouble loading. I got it loaded online,
And make sure everything nice and stable. Get there, and then they call start.
but it took a long time to process. Because it's a huge grid, right? Because you splice all of the data, so
This one you're probably gonna have to.
so I got it to load the laptop. I don't know why they printed it to look on that system.
There's a bunch of seasonable values in it.
What's that?
There's a bunch of seasonable
Two
values.
people.
On the 1 meter bathymetry, there's a bunch of sea level values sort of that you don't see when you're looking at like a 10. But when you're looking at 3D, that's why it was just things shooting up.
Making sure your heading's not trying to drift off.
So I don't see it on mine.
This is pretty
But
flat.
you're looking straight now.
Yeah,
Just
but
run everything.
see
It's
it.
pretty good for the most part. So, been running with that 85 delta.
Yeah.
I think there's like
No, I
5
don't have that.
So we pulled it up last night. It was And that was my question when they put it up here.
Yeah.
Maybe that was my.
Yeah, it started coming out of depth thing.
I don't know why it's not.
Once we started crazy time.
One option though is to clip it to just the area of interest.
Yeah, there was an app for board like foreground.
Yeah, because you have. So, he was perfect.
We are going south, right?
I mean, you could clip it into maybe 3 years and then just
I didn't turn it on. No, I turned on the other light.
stay lit.
No, it's on the
Yeah, I got it to work, so.
I do an issue push forward.
Okay. We're still playing with it, and the other one.
The next line is going to be interesting because I think there's
All of them.
a variation.
Because of what?
There's a bit more.
Oh, see the fire ones?
To the left or right of the line.
Yeah. And there's more
Yeah.
Or when we have to go to the seafloor.
Okay.
Sorry. Yeah.
Could we bring them in now and then just have them turn apart to see how they're ready to go?
Really monitor everything. Make sure you're not getting any big spikes. If you start seeing anything, especially with birds. Once you see a big, yeah. There's something stuck in you.
Yeah. The rod.
The first one.
Is that?
Tell me the whole.
Map 2.
No.
We have that
Oh, cool. Thank you. Thank
just before 10.
really sure when we were to do something.
Yeah.
After mapping, so just
Check the gauge.
Oh, I see.
how
Yeah.
mapping just didn't hold up.
I'm looking into the possibility that we don't have to recover the car anymore. What's that?
I'm happy to know that they're in there now because we have such a short time to sample after mapping. It's like any prep that you need to do, they want to be flexible here.
Had a little bit of excitement
Yeah.
today.
I hear ya. Yeah, Captain was sort of
No
looking
fancy.
into that idea yesterday, but Joe was saying it's pretty
Second
common
one, we
these
take
days.
off of it, like a career, flexing.
Right. It turns into a treatment exercise or something.
Okay, do you want to officially pass over? Yeah.
Water. Check something over there.
And
That way, it's all-
so our last
Usually
thing was
beneficial.
92, start hitting altitude
Yes, it's a kilometer. So it's,
in this one. We've never gone.
Hey, crawl and fly.
Thank you. I hate ground.
we had a few drones on board. Maybe we could.
What's that?
Okay, the drone.
Yeah.
And I've never forgiven you.
I've never forgot it.
That's a good idea.
Burning or rage or. Yeah, forward.
MTs or MTs?
Yes.
Thanks. Yep, no, it's,
How do you do it?
I'm sitting right where I'm sitting, so I see lots of seats for you.
It's a lot flatter than what we were.
Do you have a list? Would you mind? I don't know where you put it, but I'm putting together this.
Okay, so there.
I have a list of rocks that are collected, but I don't know which ones went to you. Just a list of email it to me. Or maybe you've already got it.
No, I don't. But how many accessories do you want?
So the guide member and the car and the so many.
So when I go here, I don't need to ramp up, so I'm mapping that. And here, not here. Yep. I don't get past it here.
Crystals.
7.3.
Scales and dynamics.
But this speed should be out for this thing.
So do you get the, so that's before you do that kind of debris, so do you get just the magnetization?
So that's good. It's just KSMG model, like a little bit of noise along
So I'm interested in whether the volcanic rocks, the presence of relationships, whether they're fresh volcanic rocks or different, because we have more fatty basalts and andsets that would be more of the core map. Because you can't tell the difference in the seafloor, but you have this. Where's that? Yeah, but even I can see the numbers.
What is it? Oh no.
Where, which area is this?
Open.
Yeah, okay, wow.
seawater removed. So you're left with pretty good looking seawater.
Here it's 10 meters past the waypoint, end of line.
End of line.
Line.
Yes. And just end of line 2. Thanks.
So since this line doesn't matter, I'm just gonna move the vessel and then start heading and set up for the next line.
We're just transiting now?
Yeah. Thank you.
That's it.
Yeah.
Roger, I'll do it in the waypoint for you.
Have you started the line yet?
We just finished.
2 lines.
Okay, give me 1 second.
I'm gonna just transit the next one. Is that alright?
Yeah, we might have to
Stay here.
redo it.
The line.
If they don't have it, they don't have logging on. Okay, no, we're okay.
We're okay. Okay. So I'll head up to 4 then.
Okay. So you're on line 3 of InSAS?
When I was recording this one, I'm having to do it from
So the ridge
waypoint
was
3 to waypoint 4, and then we'll map 4 to 5, or 5 to 6.
Okay.
Same line, back and forth.
Okay.
Probably you would see that, but could you distinguish that effect from alteration? So the blue area that you showed is a difference in composition. Position of clouds and reservoirs that are affected.
Do you know who started from here?
What does the reach on the maps look like? Can you see variation? Interested to know if there was a difference in all of your as opposed to, where yeah, yes, you should see that.
Pretty sure we just move into the map so you can go to that waypoint as fast as you need.
Sorry, can you repeat that?
I said this line isn't being mapped, so you can just go to that waypoint and look at that point if you want.
Okay, understood. I'll speed up.
Yeah.
Oh yeah, we're running that way.
Oh, okay.
Yeah.
Well, you don't have to worry about that stuff on left.
Yeah.
No, we're
All
going
lack
along.
of stuff.
We've got Seafloor SAS.
Looks really good.
That's great.
Across the whole sock.
Almost.
Definitely still some motion artifacts.
Yeah. That's good.
Is the motion artifact with regard too fast or too slow?
Variation.
Yeah, yeah.
And also the altitude following.
Yeah. Yeah.
I think we see sheet flows.
Oh, okay. I can give you just a little just what for any information. Yeah. See it. As long as we lie. Otherwise it'll just take a picture.
We see the sheet flows.
Okay, so if you just send it to me, so that can be selected.
Yeah, so much better.
It's incredible.
I put it in near range, but.
Look at that. But I think we see sheet flows and the ripples on the edge.
Yeah.
Yeah, yeah. Look at that. But that's the sheet flow here.
Yeah, yeah.
And it's bound by two mounds or edge or limbs. You have a bit of folding.
It's amazing. Those are collapse bits. For folks just joining us, we are still pretty early in our dive stream here. This is dive stream 611, and we are currently doing a survey at 15 meters altitude with the InSAS, which is our synthetic aperture sonar. There was a question in the chat, though, about what the reach of the hydrophone was. This is a reference to the hydrophone lander that we've got that we'll recover a little bit later today. But how far away is it, and how far can it hear?
Yeah, so the lander is about 1 kilometer away from our position now. And the ROV makes quite a lot of noise, so it can definitely hear us. The lander was deployed over some known active black smokers. So the idea is that it's listening to the sounds coming from black smokers, both the sound that the vents themselves make and potentially even noises from subsea floor, resonance from fluid flow and cracking events, that sort of thing. The range that the hydrophone can hear is something we're actually sort of testing. When we deploy the lander, and we takes about an hour and a half or so to get to the seafloor, and then we just sit above with the ship, and we turn as much of the boat off as we can. So we turn all of our sonar systems off, and we reduce power to one engine and turn all the thrusters off, so that there can be some silence, as much as we can, as silent as we can be.
Sure.
And then we turn everything back on and we go off and do something else and leave the lander. One of the things we did last night is we were doing some mapping with the ship, and one of the reasons we timed that is because as lander's on the seafloor, we drove the ship up to, I think, 30 kilometers away. And beyond 10 kilometers, we think that the noise from the vessel should not interfere with the recording of natural sounds at the bottom of the seafloor. But when we were as far away as we were ever going to get, we stopped for half an hour again and did the same thing, turned off all of our pingers and all the sonar and all the thrusters and just drifted for half an hour to again give the lander some alone time, so to speak. And so we'll see. We haven't processed those data yet. So we'll see how far it can hear us. But of course, we're a big boat. How far can it hear, a whale or a shrimp or a black smoker?
Yeah.
Again, this is something we're going to test here. We're going to redeploy the lander several times at different distances from the active black smokers, and that should allow us to sort of calibrate the sensitivity. The other cool thing about doing that is can we use a hydrophone system like this as an exploration tool for active venting? If we can figure out what sounds to listen for, and then with multiple deployments, could you actually triangulate the location of active venting? And we don't know, but tune in 5 weeks and maybe we'll have somewhat of an answer.
That'd be very cool to think about, right? Because obviously we can use like seismometers to triangulate seismic activity. And so if we can figure out what different vents or different ventings sound like, could we triangulate like what type of vent it is and where it is? Exactly. Wow.
But you brought up another thing, which is not only just where the venting is, but what kind of venting. Does low temperature diffuse venting sound different from a roaring high temperature black smoker vent at 350 degrees where you have turbulent flow and resonance in the chimneys? So maybe you could classify venting without ever seeing the vent, just by listening.
That would be very cool.
Question, what is the hydrophone weight made of, since it will be left behind? The hydrophone weight is simply a big chunk of iron. And that piece of iron will be left on the seafloor, but it'll be eaten away over time, and it'll just rust away to nothing. Similar to what happens to black smoker vents, actually. They do the same thing. There's all sorts of iron in there, and eventually they will rust away to nothing. Obviously, it'd be better to not leave things on the seafloor, but we choose iron because it's essentially environmentally benign.
Yeah.
And,
I'll stop.
I like this question of if the ROV and the boat are making so much noise that, you know, the hydrophone can hear us from kilometers away, how are we seeing animals at all? And that's something I always try and keep in mind, right? The animals that we see are the animals that have either can't move very fast, or for the fish, I mean, they've just decided, like, I'm going to stay here. But there's probably so much that just leaves. Especially, like, there's been times where we've been going over like silty bottoms, and we'll fly into a cloud, having not been through this area yet, and so something has moved.
Right.
Something has left.
yeah, I mean, that's there's many layers to that question, because it's a fundamental question of any science is the act of observing affecting what you are trying to observe. So that's a really good question.
Yeah.
But in this particular case, you know, a big, loud, bright ROV approaching you as a human, having never seen something like that before, you would probably be kind of scared.
Yeah.
I'm always surprised that a lot of these animals we see, they don't seem to care. But to your point, it's the ones who don't seem to care that we see, and we don't know which ones do care, and we never see it because they run away. So
yeah, the odd stirred up silt or depressions, fresh depressions that have like crumbling walls.
Yeah.
As you're driving, those are the ones.
Those are the ones.
Yeah.
There
Who
was
knows what
something
that
there.
was? Yeah. If someone asks, right, like, then could we deploy just a camera that doesn't make noise? Yeah. So we or you know, the marine science as a whole, there are instruments that are deployed like that. So camera landers, where it's basically just a little platform with a camera and some lights, and they huck it over the side, and it just sits there on the bottom, not really making any noise. And they can even set it so that the lights are off, and then they like suddenly turn on, and you try to catch something there. And those can be baited with food or not baited. Obviously, if you bait it, then there's a bit of a bias there in that it's drawing in things that maybe are interested in the food and wouldn't normally be right there. But yeah, all sorts of different ways to approach it. And likewise, people ask, does ROV Sebastian have a hydrophone? We get asked that every once in a while. It's like, no. We would just hear the
ourselves
screaming of the thrusters. Think about like a quadcopter. If anyone's ever been flying a quadcopter around, and we've got a 3-ton quadcopter underwater. I mean, it's noisy. It just is what it is by being a large device pushed around by thrusters. So we can't have a hydrophone because we would just hear ourselves. But we can use something like a hydrophone lander, like we are now, drop it down and move away and record the ambient noise that way. So Ocean Networks Canada has a few camera platforms that turn on for like 5 minutes at the top of the hour, and you can find those video feeds on their website. So there are some platforms out there, deep ocean platforms, because there's like the Coral City camera as well, but that's pretty shallow. But as far as deep water goes, yeah, Ocean Networks Canada, you can pull up some of their camera platform feeds. Landers are good when you're trying to either do something in the absence of the normal tech you would use, right? Like, so the hydrophone lander in the absence of the ROV or the sub, so that you don't just hear that, or a camera lander, so you can have kind of, again, the absence of that noise, but it can also stay there for a long time. So looking at long-term deployments and kind of just turning on for a few moments, grabbing some video or images, and then turning off again. Or there are like landers that have sediment traps. Basically, if you think of like a rain catcher or a rain meter that you might have out on your deck that just slowly collects rain, well, you can collect marine snow the same way, but it takes a very long time. So there's been landers that have done that, for example, as well, where it just sits down there with a big funnel and slowly accumulates marine snow.
Breeze, Rob, you can speed up just a little bit. What's that?
Before we've done all the...
No, speed up, just one more. Yeah, so what are you going right now?
No, we haven't applied all the patch tab.
What's that?
So this isn't even...
Rig, what's your speed? Alright, thank you.
Oh, it's really quiet today. So the surveying that we're doing right now is with our InSAS, the synthetic aperture sonar that we have mounted on ROV Sebastian currently. So we're only at an altitude of 15 meters compared to about the 50 meters that we were using for M3 surveys using the M3 multi-beam sonar. We're also running the magnetometer at this time, I believe.
We can measure the height.
After this, we're going to drop on down to the seafloor, and there is expected to be active venting. We've got a couple objectives while we're down there, of course, just looking around, but we do have some items we'd like to sample as well. So, potential bamboo corals, rock samples, of course, temperature, water samples in the Niskins as well. Sediment samples if we can find a good spot for the push cores. But we are also keeping our eyes peeled for skate eggs. And if we find some of those, we'll likely collect a couple, as well as take the temperature of the water that those eggs are in. And if they happen to be like right at a vent, we'll do a little photo mosaic of the area as well.
gonna get illumination from the opposite side.
Again, this is a pretty short dive, all things considered. Recovery time of 1800
Okay. There you go.
is what we're anticipating. It is currently 1300 at the ship. However, it was discussed earlier that that might change because the plan is to come back up, go get the hydrophone lander, and then get back in the water. But the hydrophone's only about 1 kilometer away, so we might just send the workboat to go get it and keep the ROV in. I don't know that there's been any developments on that plan, but John had mentioned it earlier.
Oh. 10, 20, 30.
Sure.
Yeah, we won't really know about the method by which we're going to recover, if we're going to come out or if we're going to just go get it, basically until it comes up off the bottom, and we'll sort of figure out where it's going to go and where we'll be.
Wait, that's close and this is far. Is that just a long? So we're coming for that way. What is it? Yeah, no, it's the pitch of the sheet floors. Well, this is why the ground truth these things, right? Yeah. So this is all 3 cylinder. Yeah. So it'd be darker. Wow.
Oh, well, it really depends on what decisions you're talking about being made. The dive plans get put together primarily by the science team on board, although, you know, if there's questions of can we do this or can we do that or do we need to do this option, that all plays into it, right? So that can go back and forth between the scientists and the ROV, the marine techs, the folks actually like steering the ship. So yeah, there's lots to consider, but the dive plan gets put together, and then for something like this, you know, the waypoints get put together by the scientists. They get implemented into the ROV systems, the computer for running the ROV.
Right.
But like right now I have a dive plan that I can pull up. And, you know, things change. But it lays out like what time do we want to launch? What time do we want to recover? And this one's very specific in that we're going to do like a couple lines with the InSAS. And so then those waypoints are something that our pilots also need. So included with the dive plan are some waypoints in CSV files. There's been times though where it's like pretty loosey-goosey as far as like where the ROV is going to go on the bottom. If it's kind of just like we're going to cruise until we find stuff. And then when you all were in Australia, there were some like dives of opportunity where it was like, we're kind of in this area. Let's just go check it out. But that was COVID time. Yeah. Chris got to put together the dive plans. How much of a plan was there?
Go get in the water and see what's there. Don't touch anything.
Yeah. But a lot of this planning starts, you know, before the science team is even on board, right? When they are looking at what instruments they want, what instruments do we have, what do they need to bring, and then if they want something that's going to be mounted on the ROV, we need like dimensions and weights and operating, you know, requirements of this equipment. And then we have folks that basically like pull up a model of Sebastian and try to see where it might fit. And of course the pilots are going to need to figure out like how does it get routed in. So lots of stuff. And then even then, sometimes the instrument makes it to the ship and we go, oh, it's slightly different from what we thought, and we've got to just adjust on the fly. Yeah, I should say not all of the Australia dives were dives of opportunity. Many of those were pretty extensively planned expeditions.
Basically from May until June.
Just May and June, basically?
May and June where we didn't have scientists on board.
Yeah.
And then we slowly started thinking more. There was a little bit of time.
Yeah. Well, and there were some then where the scientists were coming in remotely, basically. Yeah.
Well, and all of them, even the ones that would pretty much just go down there and see what's there and check it out, we would have, like, Dr. Beeman would be remotely on there, and he would comment, and, you know, we would see something that we knew that would be of interest to not only watching science as well, we would go closer with that. They would discuss it, what it was, what content was, things like that. We'd worked with them enough that we had a fairly decent idea of what kind of things they were focused on.
Yeah, totally. Absolutely.
Alright, that's 10 meters past.
Yep. Yep. And a line.
Alright. Just a little bit smooth.
That's line 5. That makes sense.
Right here. New waypoint bridge. Let's see how this works.
Thank you.
Yeah, our port of registry is Georgetown in the Cayman Islands, but it's not like we go there at the end of every season. If you look up like Schmidt Ocean Institute 10-year plan, we have sort of an idea of where we want to be for each year for the next 10 years. And so we're playing a regional game there where we will work within and we'll stay within each of those regions basically for the year. And so some of those regions, we're going to kind of pick like one port or two ports that we stop at primarily between the cruises. But yeah, it's not like any of those are just like a home port. It's just where we're working out of for that region.
Do you want it at 15 meters as well? You know, we had one dedicated to novels.
Yeah. Just go find Nautilus. Yeah. Yeah, we're mostly sticking to the southern hemisphere for the next 10 years. If you take a look at that plan on our site. But 10 years is also kind of a long time to plan into the future, so who knows?
Yeah. That's a different plan.
Yeah. Plans change.
Sorry.
Just toss the map up here for a moment. Let's see what we got. Got nothing. Let me get that on something. Nope, doesn't want to go. I'll keep working on that. All right, so you can see kind of the two lines that we've done. And you can make out a little waypoint off on the left side there. We're going to head on over there and then sort of cut across those 2 lines that we've done. Technically it's 4 lines. Oh, because the little...
No, we've done this.
Okay. So each of the 2 lines have been done twice. All right. Chris wants to make sure he gets the credit on those.
Well, I just want you to be consistent. This is science after all.
Uh-huh.
Is that all equally stable right now?
Yes. Huh?
I think we could just.
Sure. Okay. We're 15 meters. We're doing the line.
Yeah, perfect. Let's get the
There you go.
Not mine, 7.
7 is gonna be the next one.
Yeah.
We're headed to 9 when we start at 6.
We're on the line right now.
But we're
Taking
stable, so
advantage of the
I'd rather cut it down.
Otherwise,
Otherwise, it's a waste of opportunity.
yeah. This is how it works.
19, 17. Or you just started reflecting?
I mean, it was like a bubbling. So there's a lot of the edge that is just like fine. You'll see like it'll have like this bulbous area in the middle and stuff. Yeah, but and they've used before the left ones that are more like this to like softly grab around you and not like the slicker ones on the right hand side. So I think I would like to get like we sat down, we could, do the sheets about temperature curve in one hand and then collect the other, turn up because then we don't have to put the temperature curve down. It's just like sit down and collect the view. And then just like, we've got another one. Why does this keep happening? I don't know.
20-ish shorter line.
Oh yeah.
I don't think.
So again, this cruise is going until November 23rd, but it is not the last cruise of the year. We do have leg 2, or part 2 of Octopus Odyssey coming up as well after this. So that'll be returning back to the octopus nurseries that we explored earlier this year. The Octopus Odyssey 2 will be pretty short, though. That's just scheduled to be December 1st through the 14th. Gonna explore the area a little more, as well as collect up some of the experiments and loggers that they left behind on part 1.
still have like just one last one. Will be showtime for the bottom people, or is it the plan to recover? But one last. Yeah. Yeah. Well, if you guys find the transit level and it's just one line, I could do the photomosaic for the whole line and yeah. You don't have to come back on it, you don't have to do grid, you could just do whatever line. And that's being very easy to create. Just do it. Yeah. I'd say, I mean, if you were down at 5 meters, I'd say you'd have no problem. Yeah, but if you, like, depends what it looks like, but if you can see the features, you can map the features, like, and then, but you might as well, try and just model it as a transect, and you'll have, just make sure, I mean, I can come in here for to make sure that there's nothing in the field of view, like you're not seeing the porch, and it's all the way down, and it'll still be something. Yeah. And so you just put as much light as whatever you can see. So whatever you can see is what gets created. So if you try to blur the lights a little bit more and that, but I can stick around, like I'll be around for a bit if you're gonna find it. So what do you think you're gonna do around to? Like the long line or?
And that's the end of this one.
And the line, yeah.
Maybe the last 2%.
So at minimum, these ones. And then
We're gonna go to eventually a little bit this and then set up a little bit off of this one. So we're gonna wrap up.
okay, so not nothing.
No, it's a bit on this line.
Yeah. Okay, I'll check.
So yeah, we're gonna go rate.
But I'll keep an eye on it, and if you want to try and set up, and obviously you gotta prioritize your stuff. But if I can create it for you guys, then that's it.
Like the sheet flow and stuff, yeah.
extreme variations in the seafloor. You know, we have time.
West Edge.
No, I would do it over an area where most kids have. Before we do this one? Oh, yeah, okay. Got it. Yeah, totally. At 5 meters, we'll be able to see animals.
But I don't think that's for animals, or do we want it?
No, just to see them, just so we can see and we see them at 5 meters, and do we see them in the SAS or not?
We can try and see what.
I mean, we won't pick up animals. That's just a waste of time. Oh, hi, trees. Good to see you there.
I feel like that was intended for me.
Yes, so
Yeah. We're gonna find the durable work over.
Alright,
okay.
started.
Start line.
9-7?
Line 7.
7. Where we start.
Entry.
Did like 2 mappings to figure that out.
Yeah.
I moved that out of the way about 20 meters, so let's go right through it and I'll move it up here. So it should be the right speed.
And then what we'll do is parallel the north and south. The first line that we did.
Okay.
Just so you cover the seafloor where the SAS is already.
Alright, so just parallel
So
to
we'll-
1 and 2, 2, 3.
Yeah, and I'll give you the number of how much apart from there we need it in a second.
Sean?
Just 1 second.
Yeah, no, you're good.
This doesn't make any sense. Yes.
With the line and then the parallel line, do you have an estimate of when, the biologists might go ahead?
So we're on our last line now.
Yeah. And then we're gonna run that, trench roofing line.
We're gonna do 1 line,
And
and then we drive. Oh, my ARC project just crashed.
So
No.
line 2 meters east.
And you're gonna do parallel to that.
So we'll go north and start. But we can already find the seafloor to the north. 5 meters.
You want me to just slide or just like this?
I think 5 so how much this we but it's to see the seafloor.
Drive down here and then circle kind of this way.
and is that by like the west, like smokers?
Yeah, exactly. So this is
Yeah. Okay. And is the science role policy, like X amount of time?
I'm gonna, there's all sorts of groups. I've looked at the bridge and figured something.
But as of now, out of order is still 5, right?
As of right now, that's correct.
So that would be required.
Yeah, so there's not a lot of time.
No.
3.
Well, it's not 2 hours coming up anymore. Or is it?
No, it's not. It's like an hour. Let's see what I have. That's 6 o'clock. So
we have to find it in the lights.
No, we do. It's gonna be better. No, it's gonna be a sunset recovery. Just because we like the drama.
I'll make a long title.
Yeah, because of how low it is.
Okay, so if it's an hour and a half, we're looking at like a 4:30 or
4:30 on the morning, so it's business 3 hours. We're gonna come back.
Yeah. I know. I'm really hoping just for like an egg or two to not have to process lunch for the first time. Keep it slow and just. Does that make sense?
No, you want to be maximizing it.
Okay, here's where you can just follow up.
We're gonna filter it.
Well, what plan B is to do it slowly and A will just be torpedoes. Okay, thank you.
So 2 is a okay number?
Well, yeah, just like anything's kind of get me started,
I mean, I kind of think that if you see it, you stop. It's something that has to be done.
Yeah.
There's no point for setup. There's no point. Well, let's go back to normal.
Yes. And I'm hoping that we set down a place where there's actually a compass. So it's like with the right arm, temperature measurements, with the left arm, all in its box. It sounds like a good plan.
Looks like a great plan.
Bang them out. Actually, we'll do the 2 operations at the same time. Yeah. Okay.
Dual armors.
That's what we're talking about. Alright.
Might need to make it or
It's fine.
Very cool. Get the texture chicken. Yeah. Great image. Very bumpy seafloor though. Yeah.
Okay, how's your day?
Turns up.
Yeah.
Hey Alex, in your answer about the decision making, did you describe us as an autonomous collective?
No.
No? Dictatorship?
I mean, I see you working over there, but I'll toss it your way if you think about it. But like, I guess what's your experience of organizing something like this from your side? Like, what sort of things are you looking at, and what, like for us, right? I kind of said there's like a lot of back and forth where you come to us with sort of what you want to be doing and like how you want to approach it, and the pilots might go like, it'll work better if we fix it on the ROV this way or something. But like, you're balancing a lot of different areas of study for this expedition. So what does it take, I guess, to kind of make sure that all melts?
Yeah. Let me think about that.
Yeah, no, that's why I was like, if you got time. If you got time.
Yeah, we got time. Yeah, that's an interesting question. My answer today would be very different from my answer from our first dive.
Sure, yeah.
And the reason for that is because, as you mentioned, we're doing a lot of different things on each of these dives. There's the mapping, and in some cases there's two different types of mapping, and then there's the work we're doing at the seafloor, which is a combination of collecting samples, and then there's all sorts of different kinds of samples we're collecting, and then there's just the photomosaics, those surveys, and then there's just the looking around exploration aspect, which often gets sort of pushed to the end, yet in many cases is one of the most important aspects of a dive at the seafloor. And we've been having some discussions about this. If I, given the liberty to oversimplify the sort of geology-biology rivalry, that often friendly rivalry for us at least.
No one's dead yet.
There's, the biologists, I use that term in the broadest sense possible, want to collect animals. So it's a physical collection, and then you want to understand the immediate environment in which that animal was collected. And so you look around a bit. And for the geologists, we do something very similar with a rock. You want to pick up a rock, and then you want to understand the immediate environment of that rock. But in both cases, it's important to understand the greater environment, the greater surroundings, not just what's in the field of view of the camera, but what's 100 meters away or even a kilometer away. And I think it's important for both the biological and geological perspective, but it is more ingrained in the training of a geologist, I think, to think at those greater scales. Now, I'm painting with a pretty broad brush here, but if we accept that, then one of the things we ran into early on is we'd get to a new area and we knew there's a lot that we wanted to see in this, whatever, 500 meter by 500 meter box. And so, okay, well, let's explore, let's see what's there. But as we're driving around, suddenly a coral appears out of nowhere, and we're like, oh, we need to sample that coral. And so we stop and we sample that coral, and then, okay, let's keep going. And then we find something else, another rock or another animal, and like, oh, we better stop and look at that. And that process meant we didn't move very quickly, and we weren't covering as much ground as we wanted to. So what we've changed is that when we're down at the seafloor, one of the first things we're going to be doing going forward is not sampling initially rocks, biology, fluid, all of those things, and simply drive around and see what's there. And especially for the geologist, that is a lot of information. Some could argue it's even more information than the rocks we pick up. That geological context simply from observation of the seafloor over a wider area. So what we have to do is try to balance that with the needs of everybody. And we have, I mean, if I look at the dive plan, you know, in terms of sampling, we've got 5 objectives and then several sub-objectives under each category. And so the art is trying to balance that so that everyone gets what they need.
Yep.
And that we do it, sometimes it has to be done in the right order, sometimes it doesn't matter. So that takes planning, that takes a lot of discussion before the dive. And then you get to the seafloor, and then it's never what you expect, and everything's different, and those plans just... And you sort of make it up as you go, and then you find something that you didn't expect, and suddenly you've spent 2 hours, which is important because it's something new and potentially a new important contribution to science, but at the same time, people have projects and theses for which there's very important data that has to be collected.
Mm-hmm.
So that's a really long-winded way of saying a lot of communication and a lot of trying to make sure everyone is getting what they want.
Yeah.
And there are people who are vocal and there are people who are less vocal. And so we're always trying to nudge the people who are less vocal, or the people who, you know, are a bit too timid to be like, I want that thing, I need that thing. And but we're all, so far as I can tell, mostly reasonable people on board, more or less. And I think
that's an answer we'll come back to.
Yeah, okay. Anyway, I think it's been working out really well.
So a lot of flexibility required, it sounds like.
Flexibility and patience, yeah.
And so going like way back, right, before the ship, before even, you know, like you give this like proposal basically to SOI, like how do you start thinking about an expedition, like at the very start? How do you consider what aspects you want to cover and make sure that you're bringing in like a widespread, I guess?
Right. So I'll tell you the history of this project because we've got time to kill. So we submitted our first proposal to Schmidt Ocean Institute probably back in 2017. And this was my first kick at the can of being the PI of something of this scale. And so I knew what I wanted to do, and we'll come to the science in the area in a second, but I knew what I wanted to do. But you always, for any of these expeditions, you put a team of people together, and you don't just do one thing. You do a lot of things, because the amount of resources and money and time you want to maximize, you want to get as much science as you can out of an expedition like this. And we all have our collaborators and people who do different things, but things that are linked to what you do. And so you put a team together. Generally choose people you like, because you're stuck on a boat with each other. And so anyway, we did this, and our original area of interest was a place called Southern Explorer Ridge, which is north of the Juan de Fuca Ridge, and this is a series of mid-ocean ridges off the west coast of Canada, so in the Northeast Pacific. And it's a fascinating area because the Juan de Fuca Ridge has been significantly explored. There's hydrothermal systems there, and then there's a big transform fault, and then there's Explorer Ridge, which ironically, despite its name, has received far less attention and exploration. And geologically, this is one of the most insane places on the planet, because you have a nearby hotspot, like Hawaii, Iceland kind of hotspot, and you have mid-ocean ridges, a couple of them that are offset, and the whole thing is being subducted underneath North America. So you're subducting a mid-ocean ridge. There's sediment coming off the continent, so half of the ridges are buried in sediment, which means biogeographically and geologically they're very different from the ridges that are further away that don't have the continental sediment influence. So it's a dream for a geologist, and for the biologists, it's similarly fascinating. Canada right now is trying to turn that whole area into a marine protected park, just because of the geological and ecological insanity. Anyway, so we had several scientific questions trying to understand how different vent communities are connected or not connected within this region, and then the geodynamics of a subducting mid-ocean ridge. What are the hydrothermal systems like along there? And so we put together a project that included both geologists and biologists, all associated with hydrothermal vents. And then this was, of course, the area where we also wanted to test the synthetic aperture sonar. It's an environment that sort of geographically is very similar to where we are now. So that was the project, and we submitted it, and it didn't make the final cut that first year. So we learned a few things and tweaked a few things and had some really great feedback from reviewers. Tried again, got closer, but again, not quite there. And then the third kick of that can, which was 2019.
Folks can see what's coming with,
yeah.
With that.
The project was successful.
Yeah.
So 2019, Schmidt Ocean Institute said, yes, we are willing to give you ship and ROV. And of course, this was back with the original Falcor. And we're going to have an AUV, autonomous underwater vehicle, for all of the mapping. And so the project was going to be two legs, because it was going to be difficult to fit both the ROV and AUV on the original Falcor, which was a much smaller vessel, especially the back deck. And so, so that was scheduled for 2020. And, what could go wrong? So, of course, 2020 comes along, pandemic, and certainly no one was going out to sea. And so the project was put on hold. Most people weren't going out to sea. And of course it was during that time when Schmidt Ocean Institute made the decision that they were going to replace the original Falkor with this magnificent vessel. So the change to the Falkor 2, of course, not an immediate process because, I mean, how long was the refit of this vessel? Year and a half?
Roughly.
So,
Roughly, yeah.
right. So the vessel was refit for a year and a half and turned into a research vessel. And so that plus COVID added delays to when the project would take place, but the new vessel was in the Atlantic. It was being refit in Vigo in Spain. And so Schmidt Ocean Institute asked us if we were able to move our project from the Northeast Pacific to the Atlantic. And for the technology aspect of the mapping, that was an easy thing to do, but of course for some of the more scientific questions, it wasn't a simple thing of, we no longer had a mid-ocean ridge being subducted. That doesn't happen in the Atlantic Ocean. But a lot of us had worked in the Atlantic, and there were hydrothermal systems there for which we could answer many of the same questions and test the technology, so the project was moved to the Atlantic. And we were scheduled to be the first project on the new vessel, but as is always the case, a big project like refitting a vessel takes time, and it took a bit longer with COVID, and so there were some challenges associated with that, and so our project got moved from, it was supposed to be January of this year, and it got moved to later in the year. Of course, later in the year meant that the vessel had gone through Panama Canal and was now back in the Pacific.
Yep.
But not in the Northeast Pacific, in the Central Pacific. So we had to move the project again, but at this point we're getting good at this. Again, we knew that we could test all of our mapping technology out here, and there are a lot of hydrothermal vents out here that we could answer a lot of the same geological and biological questions. So there's quite a history to the project, and as the project evolved, the team evolved.
Sure.
Some people who were really interested in the first project of the Northeast had less interest in other areas, and other people came in who had more interest, and so the team we are now is partly the same but also very different looking, and that just evolved organically, word of mouth. Oh, now we're going to this area, and the team needs certain expertise, and other people said, hey, I'm interested in doing this one thing, can we work together?
Yeah.
And so where we are now is very, very different from where we started, and that's okay. There will be other opportunities to go back and try to do what we originally planned to do. But you just, these sorts of expeditions are not easy, and a lot of things have to align in terms of resources and schedules, and in the end, here we are. The big difference between this project and the original one is here we don't have an AUV, we're using an ROV instead for the mapping, which has worked out wonderfully, and I'm actually quite happy that we've had the opportunity to do this. But the original plan was the SAS mapping and some of the high resolution multi-beam and magnetometer stuff was all going to be done with an autonomous underwater vehicle, which is really what the InSAS was designed for.
Yeah.
I mean, Caroline was just showing some of the initial results that we got on what we're doing right now, and it's pretty mind-blowing. So stay tuned for more of that.
It's interesting to think about the difference in approach there, right? If you had an AUV, obviously you can kind of like huck the AUV out and say, hey, go check this out. and maybe go do something else, but you don't know what you've found with that AUV until you recover it. Whereas we're right now, because it's on the ROV, we're able to like be processing it as we're going.
That's right. That's one of the huge advantages, because we've been making all sorts of adjustments on the fly to how we're collecting these data. And I mean, we've learned a ton. What we're doing now compared to how we started this is completely different. It would have taken us significantly longer if we would have to wait for the results, process the results, come up with a new plan, test that plan. We'd be weeks behind. So I think this was a blessing in disguise. You know, the AUV thing with the changing schedules, and it just didn't work out in the end. And I don't know. That's okay.
Yeah.
Thanks, guys.
Got a little bit of a pilot watch change happening here. Jared and Carl are jumping in. Yeah, I think it's been, or you know, it's an interesting idea of like go through with the InSAS or even the M3, see immediately what you've got, and then drop right down onto it and check and be like, yep, that's what we thought.
Yeah, I mean, that's the dream. We were talking about this, you know, for the viewers at home, it's probably a bit frustrating. Our earlier dives were 12 hours of just flying around, you can't see anything, and then finally we get to the seafloor. And, I mean, we felt the same here. But in terms of, you know, all that flying around, we produced these very high resolution maps, and we had that information right away. And then we go down to the seafloor and we know exactly where to go. Exactly. Because we could identify the features we want, you know, the chimneys and the volcanic features, you know, collapsed lava flows, all of these things. We can see those in the data. And so we're spending half of the dive on the seafloor instead of all of it. But I think because we have that map, we accomplish just as much, if not more, in half the time. Mm-hmm. Because otherwise, you're driving around looking for things.
Yeah.
And that takes a lot of time. And you don't know what you're missing. So to me, having this ability is something I'm going to want every ROV dive. If the data has not already, so where we are now, the data, we have high resolution bathymetry that was collected with the Sentry AUV years ago, and those data are available, so we don't have to do that in this case. But if you're in areas like we were before we reached this area last week, you know, we didn't have that. We're able to create it right away and then dive and see exactly where to go. I mean, that's so much more efficient.
Yeah, I think even if you had an AUV that could be mounted with this, it's almost a thing where put it on the AUV, drop the AUV off, go to another site, have an ROV that still is equipped with it, and start a process like this. But then for your next dive, you'll just have whatever that AUV gave you.
That's right.
Yeah.
Yeah.
5 meters?
Yeah, I mean, still having an AUV is still incredibly.
Sure, yeah.
You know, they're faster, and as you said, you could be multitasking.
Yeah. But for the approach of the ROV, being able to map out the site immediately or before you dive down is really nice.
Yeah.
Not yet. I haven't started yet. I'm still going to the...
Are you needing this on ASNAP 1 for this, or are we doing a photogrammetry? Okay.
And this will be going downhill, correct? Or uphill?
Should be pretty flat. Hold on, I could check.
Downhill? I can change it real quick.
Which direction are we? We're going north.
Currently I'm going north, and then I'll be setting up to do a transit south.
Okay, just let me know before we start that, and I'll kick over to ASNAP 1.
The transect south is pretty flat until you hit some big drop-offs.
So I mean, that's just up here that it would be. I'm going uphill right now.
There's a question, who is we? How many scientists work on one proposal?
Wow.
That's a good question. When I put the first proposal together, I think there was about 8 co-PIs. And that was a mixture of geologists, biologists, mapping experts, again, all of the sort of main fields of study. So 8 of us, mostly from Canadian institutions, but we had people from the US and the UK and Japan, and then again, as the project evolved, people came and left as the science changed. Oh wow, look at that.
That's cool.
Wow.
So pizza oven.
Yeah, little pizza ovens, basically. Yeah.
Then it would cook pizza pretty quickly.
They said get it as hot as you can.
And y'all want me to film the opposite way, right? Y'all are still down with that?
Did you hear J-Ross' question?
That we don't want to fly the other way.
Are we doing a
y'all
reverse
want me
line?
to
Yeah,
go-
we're going north and then
Then,
we're doing the photogrammetry from north to south.
okay.
And that's it.
Which is going to be like pretty much in this location. That's a good corner, right? Well, that's cool. That's right on the back line. Not have anything to do with this. Just want to see that view we're going to have.
So right, so we is generally mostly scientists, professors at universities and government research institutions like the United States Geological Survey or Woods Hole or National Oceanography Center in the UK, JAMSTEC in Japan. And then the actual science party is a combination of some of that group of PIs, and if those principal investigators don't come on the cruise, they have their students and postdocs and research associates. So our group on board now consists of, I don't actually know the demographics off the top of my head, but probably graduate students make up more than half of us.
Can you put the laser on?
And there's a good number, maybe 4 or 5 professors or senior scientists, and then some postdocs. So a range of career stages, I guess.
So you said this was the first time you were applying as the lead PI.
Yes.
So you'd been on cruises before then, not as lead PI. Doing the actual process, is there anything that was way different from how you anticipated?
Oh, it was chaos. Yeah. I think that this is probably cruise number 23 or 24 for me, so I certainly had enough experience to understand what goes on at sea.
Sure, yeah.
But that doesn't necessarily prepare you for the planning process. It's kind of like when I have in my lab, back home I have a gamma spectrometer, and for my PhD I used a gamma spectrometer. But going from a gamma spectrometer user to a gamma spectrometer owner, a heck of a learning curve.
Yeah.
And this is very similar. Like there's issues and there's all sorts of different parts of the planning process that, as a participating scientist, you are not exposed to, but as the chief scientist, you have to know everything. Things I didn't even know existed that you had to know. It's sort of the unknown unknowns and the known unknowns and the all that stuff. So it was a learning process. I have colleagues who have done this before, who I relied on for help. The Schmidt Ocean Institute were incredibly adaptive and understanding and supportive, and that of course makes a huge difference. And so, with the help of many, here we are. But there were certainly times that I felt completely overwhelmed, and but people were always there to step in, and I think next time it'll be easier. Someone asked me if I would do this again, considering all of the go back into the history of the project changing and changing oceans twice, changing a ship once. I've generally worked in international waters, and now we are working not only in Ecuadorian territorial waters, but in the Galapagos National Park.
Yeah.
And so there are rules that come with that. That's the whole point of a national park. And so that was an added dimension of permitting and contracts, and so that was an additional challenge that I did not originally foresee.
Yeah.
Again, there were people from the park that were able to help, and Schmidt Ocean Institute was able to help, and so it takes a lot of people to put this all together. And
could you drop a marker, please?
sometimes I just stand back and get out of the way. And,
A lot of moving parts.
A lot of moving parts. And it continues even while we're at sea. There's still a lot to be organized.
This whole ridge right here is full of coral, just so you know, like everywhere.
Oh wow.
This little one you see.
That was just the first Pixar that we've seen.
Yep. And then I got more up here. Was that bubblegum?
No.
Or Swiftia?
Swiftia, I think.
Someone's curious, what is a gamma spectrometer?
A gamma spectrometer is an instrument that measures natural radiation coming off of objects. In my particular case, we measure gamma radiation that is naturally emitted from rocks that we collect from hydrothermal vents. And we use these naturally occurring isotopes to date the rocks using principles of isotopic decay. And so this is how we are able to determine the age of a vent field or the age of a chimney. It's because of these radioactive isotopes. In this case, it's a radium-226, which is an isotope that has a half-life of 1600 years. There's a whole suite of different isotopes you can use: thorium-230, lead-210, and so a gamma spectrometer simply measures the radiation from which we can calculate how much of those isotopes are in the rocks.
All right, I'll kick over to ASNAP 1 here.
Might take a second to set up life or just go through the checklist if you want to save a couple of photos.
So this is a spectacular example of a fractured It's like an old lava lake or sheet flow. You can see some texture on the surface there. Dusting of sediment, so we know it's not brand new.
Alright, what's so do y'all wanna come look at my lake, see if y'all are happy with what I have? I think I have.
5 meters? Yeah. Okay, great. So if we can come down to the altitude that we think we're gonna fly at.
Okay.
And start with checking the science and tilt it down. So I see that guy, so if we can tilt past, then yeah, that's great.
Lasers on, right?
Yeah, lasers on. So if we log this as a photogrammetry transect, and then if you put in the comments that it's ground truthing for SAS. Are we not at 5 meters here?
Yeah, you gotta understand there's a crack underneath.
Oh,
so
okay.
you really gotta go by the
Yeah,
image.
yeah.
You go by altitude.
Yeah. I was gonna see if when we got to 5 how it was.
Yeah, it,
But
so roughly about 5 meters.
okay.
According to the terrain, which is
Yeah.
just changed up. Crack.
If you hold it there, do you need to be higher to get more area to ground truth? Because we could go higher for the purposes.
So for me, the more terrain we see,
Yeah.
the best it is. But
Okay.
then for your photogrammetry,
It'll still work. So let's go up, and I'll see. So if we could come over to the bottom a little bit more, and then we'll find a new.
Not sure if we dropped a marker at that first one when we first touched down, so we'll see. I know we dropped a marker at least one other coral as we were going, so it's possible.
Hold that here. Is it good?
Yeah, if we could hold it here, and our lights, are we doing that like throwing the top lights and turned off the bottom lights? And if not, can we do that still?
You want to say that you want to turn off all bottom lights?
Because usually we turn off the mud lights, if that's right, and then.
Which gives you a shadow down there.
Yeah.
Yeah.
We haven't done a photogrammetry this high yet, so it might be a little bit of playing with it and figuring it out.
Floods.
Okay.
Oh, that's
and
good.
if it's floods and muds.
Muds. Yeah, muds aren't going to make a difference unless we're mud.
Okay.
They
So
will get rid
that
of some of the
corner-
lower shadows.
So that corner will stay there because of the height that we are, right?
Yeah, let's see if I can. Washed out.
Yeah.
Yeah, unless I turn off some other lights and open up my iris and ISO.
Do you mind playing with it a little bit to see if we can get rid of that corner? And I don't know if that corner in particular is dropping away from us right now.
I think it's dropping away. I don't think it's a lighting thing.
Increase ISO. Lower destroy. So you want more light anywhere?
Even light, but I would lean more towards even lights than like hot spotting anything. Like I think I could actually work with this. Yeah, if you're happy with this, I can work with this.
yeah. And you're using the screen on the right.
Yeah, this screen's the same as that screen. That's the one we're using.
Is it possible to tilt up the screen in front of you to now?
This one?
That one?
So that
Okay.
Oh yeah, I can change it. Are you wanting to use that light?
I want to use it to just see a bit further away.
Well, yeah,
But it's not-
I'll typically be using that as I'm flying, so I'm not crashing.
Okay, perfect. Yeah, that's great.
Ooh, nice one. Okay, so just checking, we've got the lasers on, we've got our field of view set up, and then the last thing would be A snaps, and then flying speed, you know.
But have not.
Yeah, I mean, half knot is generally okay for me, okay with
Whatever
you.
you need.
Yeah, great, then I think A snaps on and we're good to go.
Roughly this view right here.
Roughly that view, yeah, that'd be great.
All right.
A snap is on?
No,
No,
we
it's gonna
want.
be okay.
Yeah.
Yeah.
Do you leave the lasers on the whole time?
Yes.
slide it over to the sun point.
Or do you see lasers in there?
The program takes that.
Okay.
Now, do you want to do it right over this line? Because this is going to be a gap, right?
No, we moved the line 90 meters east.
Oh, you moved the line. I see.
Yes.
Okay.
That's
All right. Punch it, Chewy.
Okay, so start of. Yeah.
Okay, I am.
Who's annotating?
I am ready to go whenever you guys are. You want to start the vessel?
Good to go? Good.
Yeah. I can do it.
Nice.
Good to go.
Alright, let me just get the vessel going.
And then if you just write a comment about line number and then.
Right, Charlie,
name,
got an
and
update this way, Mike.
speed between
It's a
half
long
knots.
one, if we did, I'd have it on, that'd be great.
Yeah, on that.
What did you call this line? 9. Okay, so line 9, not 11. I know the waypoint's 11, but they're line 9. Yeah. And then maybe once they get going. Yeah, maybe you can put down general altitude and it's going to fluctuate, and then speed is 0.5 knots. I know you'll like have total knots every time. Meters per second.
Speed up on there, speed over ground.
0 right now. Okay. Well, maybe you could just hold that until they start to achieve double nice but it's not.
Okay, I'm just waiting for the vessel to get going. Alright, that's me starting.
Thank you. Jared, can I ask you a question? Did we say 0.5 knots or 0.5 meters per second?
We told him knots, and I'm going 0.2, 0.3 meters a second.
Okay.
So half knot for both of them.
Okay.
That's what y'all been doing? You want to go faster?
Yeah, it's what we've been doing, but at this height we could go faster. So maybe we could bump it up a.
We told him to go 0.6, or even faster than that.
Maybe we could try 0.7 and see how that goes. Just things aren't whizzing by us when we're this high up, so I think it'll be okay, especially at 1 second.
Roger that.
Thank you.
Out of focus might chase if we
Yeah.
go too fast.
Speed
Okay.
up to 0.7.
Yeah, so we'll see how 0.7 goes, and then if we have that, we can dial it back.
Can you repeat?
Yeah, if you could speed up the move to 0.7 knots.
0.7, that's a lot.
You just have to go a little bit higher, like, basically it's what we see. Yeah. That's right. Yeah. Well, while this is running, for the next thing, I'm gonna grab my computer so that I'm here.
Mhm.
Murky there.
Yeah, some fire over there. I really thought it wouldn't be that kind of- We had the same one. Oh, I used to. One day we'll get that one.
That's the noise it makes when it's doing it. Okay.
What are we doing after this?
We're gonna head down right into the axis and
Go play with some vents?
go play with some vents. Maybe pick up a coral or two on the way.
No good distance from bottom?
Yeah. I'm good with that.
Just let me know if I'm drifting too high or too low.
I'm gonna talk to the bridge again about hydrofoam.
Well, we're driving south. We're driving north. So we'll be like, like 600 meters away from Friday. It'll be tight.
And just so you know, this is the stability at going 0.4.
This is a what?
This is going 0.4.
Yeah, I
So,
think that looks good.
so just so you know, like, you wanted to go a little bit faster.
You might go faster?
No, for InSAS.
Ah.
This is,
Oh.
you watch, I'm going 0.4 instead of 0.3.
Okay. And it's stable?
Seems to be. But I'm also not locked in auto altitude, so
Okay.
I'm not hitting it as hard as the auto. Out of depth. The heavy just did a little, nothing drastic. So I think we're just kind of up where we're getting a little. Let him go and tell me.
This looks like a lot of setting, but
Okay, so I just talked with Pete, and taking the workboat's not on the table. So, 1,000 meters away, why don't we just drive over? Does that make sense? Like way better use of time than up and down.
And then we're gonna wanna be in that area for. the
We
next
could
thing, or
just
we're gonna
we
drive
could
back?
just be in that area because there's more to do there. It doesn't fit my original plan of sort of working our way west to east, but that's just my OCD talking. So So Iguanas east is 1,000 meters away, and the lander, its current position, is another 200 meters beyond that. So I think Captain wants to get it out of the water as soon as possible because of daylight. So we'll just drive, and if we could drive along the seafloor, that's... So what that means is we're not recovering then at whatever time I said, 6 o'clock.
Just start driving over there.
We're just gonna fill up the ROV.
Do you have any mapping between here and there?
I mean, we could run no. I'm just thinking of running an InSAS line, but we'd have to do that slowly.
Yeah. We can try 0.4 meters per second.
We could do little windows.
So, how fast are we doing it right now?
Right now, I've been going 0.3, 0.4
Meters per second or knots? Meters per second.
So right now we're.
Okay.
So that's what's going on.
I gotta put my math hat on here.
That's what's currently going 0.7 now.
Okay. Let me do some math.
Complete.
Here.
And then we go to the southeast, correct?
Then
Am I driving? Let me see where we are here.
Sorry, so doing more is now?
If the transect is complete, we could turn it off.
Yeah, maybe around, just 180, 180. And, are we done here? Is that, we're heading down
All
to
done.
the, okay.
It's rugged.
So, we're right there. So about 300 meters. We're good to go.
Good to just.
Let's just do the same thing, lots of seafloor. If we see a coral, we will stop and pick up a coral if it's a bamboo.
Okay.
Is a coralologist
Not here.
not here? Oh.
John, is it safe to zoom out on the nav there or on the little mouse, or are we gonna?
Yeah, that mouse right there, if you, the scroll should take you where you want to go.
Hang on. We're not going to another galaxy to do this.
That's fine.
Okay, good.
Here.
So that's, wait, don't go away. Come back. That would be a 50-minute line if we sassed
now?
one line. No, not now. To the hydrophone pickup spot. Now, is that a good use of time?
Yeah.
Quite late.
So it would be. I mean, we'll be here, and we need to get here.
Yeah.
So we could either scoot up there and do something like that,
We'll map the direction.
or I mean, we could go straight down center lane.
Yeah. And we pick one side and we move along it.
I mean,
So
we
then
could. We could pick this, well, we have to end up over here, we'll be here. We could pick this line and we'll be shooting that way. Sure.
Okay.
I mean, but we're shooting, I mean, this is not a insignificant cliff right here. In fact, it is a 15 meter cliff, so you'd be shooting down off of that.
Yeah, we had worse.
Yeah?
That was rejected.
All right.
Otherwise you can go stay along the south fault.
And shoot
Which-
the other way.
Yeah.
I mean, it's the same thing over
Topography-
here
The
though.
topography rises more.
That's 10 meters. We would be able to see, if we did it down here, we'd be able to see these guys. Those are vents.
Yeah, let's do that. And on the sea vents.
Okay, so we'll do it like this and then come up here.
Yeah.
And that'll take about an hour. So 6 o'clock, so 5 o'clock we have to take off. That gives us just under 2.5 hours to poke around. Alright, that's the plan.
John,
Did anyone write that down? Because I just forgot everything I said. Yes.
should we be flying a market?
I thought someone went to get her.
And then, could you tell us where we're heading, what waypoint?
We're heading here, and then we're going to start working our way this way. And there's vents, all sorts of science in here. And we have 2.5 hours, and then we're going to
We have the waypoints in there, if it's time to throw out like the geology and biology waypoints.
we're gonna do a line of sats all along here.
Yeah.
And then we have to be here at 6 o'clock-ish around here. The ship has to be here. The hydrothermal.
And are we gonna try staying in the water for it?
Are we gonna try seeing the what?
In the water for it, or do we have to recover as well?
No, we don't have to recover anymore. So this dive is gonna go on until the ship runs out of fuel.
No, but I got emails. Is that news back from
Yes. What would you like from me at this point in time?
Well, the only thing we need to do here besides look around as we get towards the axis is come close. So.
So do you want me to bring it when I see it? Like we just passed over very close.
Oh yeah, you need to be like okay, that.
Okay.
There seem to be a lot of them. So.
If there are a lot of them, I can choose a site where there's two things at the same time. Like if there's enough to be selected.
Right. Well, I don't know if are we going to be passing over?
There's one right here.
Yeah, that looks like a decent size.
Can we take a look at this column?
one hanging from there?
No, this one right there, the sticky one. What was our conclusion about taking the whole thing?
We said yes.
Yes.
Okay. Let me look at how thick the base is on this thing.
Yeah. So
Okay, it's still not very thick though, so.
So you don't want a sample of this?
Deciding.
Okay, yeah, decide quick, so I don't have to stop the vessel, but.
Yep. It's very, it's, how tall is it? Can we get a zoom out?
Oh, that's tall.
Very large.
If we're taking a whole
Yeah.
thing, maybe we should.
Mine?
Yeah.
Okay.
Okay, keep going.
Let me get ahead real quick, and that way, if I do wanna get one, I will be in a better spot.
The thing is, though, is it going to be the taller it is, the thicker at the base? Like, are we sort of,
Well,
or is that not a?
like I think the general idea is bigger, taller, bigger
Yeah.
base. But like that one wasn't even a centimeter, so like we're not gaining much by going taller. You know what I mean?
Yeah.
So I don't really know. So, John, I think this is where I think I need a little bit of help from a map and like I don't know where we are. I don't know where we are relative to the lines or what distance we're at right now.
Here.
Okay.
See this?
No, I get the flater mouse. Can I still flater mouse?
Sorry?
You're looking for something like that, that's bigger.
Potentially bigger base or like the same size base but just like this.
Okay.
I have us 500 meters away from the coast.
This is where we're going. We're right here. We're going there, and then we're going to be heading into here. And this is where the vents are. In here. So I'm thinking if you want to get a far view, this will be our opportunity.
Right.
You're 500 meters away from the closest veins identified by anyone else.
Yeah, so maybe let's just stop at this one and pick this one up because if we want to get a background one that's far away.
This one here?
Yeah.
Okay, we're going to.
You could ask them to that those waypoints are the waypoints that we created with our labels, so that you could have them on here. It's just not checked right now.
Yeah. I don't remember anything being on this side unless I'm
We got.
incorrect.
You mean like
Where do you want to put it?
This is going to go in a bio box, the back one that's subdivided.
You want the full thing, right?
Yeah, can we just check what's on the tip there? Okay, it's just folded over. Yeah, let's just take a scan down the stem and make sure that there's nothing growing on it that we're not allowed to collect, just really quick.
you know that anything we collect, like we want supposed to collect, we can just preserve and give to them. They're actually even more collections from us.
Okay, okay. I did not know that.
Yeah. There's an acorn worm, is that right, in the background? Yeah. Yeah, but Stuart mentioned it when it's here. Anything we get, and especially if we see anything interesting that is on our list, we can collect it and we keep it and we cover it under our list. It's not cities.
Yeah.
Sorry, trying to get focused. Yeah, they know. Alright, you want me to go back to the top?
Yes, please.
Yes, please. Oh, it's the Scott lobster. Okay, cool. Yeah, let's go ahead with this. Let's get a picture at the base of the lasers.
That our bio one is loaded up here, that if you wanted to have it on the map, you could.
We could put it on. I've got it on my screen. Although I don't have a key.
The key was up here. You take a look at where it is.
Yeah, that looks great. There's lots of
perfect.
nodes, which is awesome. Is this gonna be, oh,
Wait.
could I get with the lasers at the very base of that rock? Is that possible? I know this is asking a lot, sorry.
It looks thicker than the previous one.
Yeah, that's pretty good. This looks great, yeah. So if we can grab that at the base, as close to the base as you possibly can, and then, yeah, we'll try not to break it, but obviously it's quite large, so. Is this going to be the first bio sample?
Yes.
She still wants to see.
So I have S611 bio 1.
Yeah. Control yourself.
You'll get that. You had them that way around.
But the sequence question, I can't answer it. 'Cause I don't even know
Didn't get this.
What's that?
Where are we putting this at?
No.
John?
In a free fly. Yeah. Sorry, what?
Bio box, which bio box do you want?
Oh, one of the back ones,
A big one?
3 or 4 big one, yeah. And whatever. So these are all just markers, points of interest. They will just
Just record wherever this is.
as we go.
Is there a piece of paper? Oh, here it is. For the numbers, there's the,
So whatever one you think is easiest to stuff it into. Doesn't matter. They're all open.
the key.
Get off.
Do you like 3?
Yeah.
A little bit more room to work.
This was right location. Oh, right.
Top left box.
Maybe 3A.
3A.
Who's logging?
I'm doing the third one.
I was just about to log the sample. Have you done that already? Okay. Just let me know. For sure, sorry. Yeah.
So we need the person who's on watch.
I know.
Who's the logger to be logged?
I'll back off. I just didn't think anybody was.
You're far too quiet over there.
Come down. Nice, and you can start working your way. Push it in.
We're okay, I'll back up from here.
Really starting from. Come down a little bit.
Cool.
So the only risk of doing that is just that that might cut off that base even more.
Huh?
Like the way that the lid just closed, would that have?
It's pushing, the lid pushing down on it like that.
Yeah. Okay. So the stem isn't sticking out at all?
No. It's, the lid held on, you caught it.
Okay.
And then went down.
Okay, cool.
If you look in a minute cam, there's nothing coming out at all like that. It,
Okay.
Oh, I see. Yeah.
alright.
Now when we open it to put other things in there,
We'll cross that bridge.
Yeah. Let's just ignore that for now.
am I good to keep moving?
Keep moving south, yes.
Yeah.
Bridge ROV, can you on that move please?
Yeah, I'll keep on my favorite.
Thank you.
Is this still always snap one?
No.
I want that to be snap one.
So I'll go another 100 meters or so south, and then we'll head east.
It's a beautiful mushroom coral.
Wow.
Yeah, it's stunning. We won't collect it, but it's really nice. It's beautiful.
So what can and can't we collect then?
So this one technically can be collected. It's just that they are pretty, like they're a bit harder to sample because they suction up into a ball, and they also don't have a like calcite skeleton, which is another thing that a colleague of ours is looking for in these samples. So I'm going to be sharing everything that I take with them. So it's better to take something like a bamboo that satisfies both of our requirements.
Is that another black?
Yeah, that's a black, yeah.
We can't collect black, so we're just not collecting black corals?
No, so black corals are under CITES protection, so we don't collect any CITES corals yet.
The marker stack.
The sample? Or the older one, the reviews? One that had like the big ball on it? The mushroom coral.
The one that we also discussed. The one that
Is that a Gorgia?
But it's beautiful.
Yes.
I like that.
Mushroom.
It's a Gorgia? Gorgia?
Oh.
Yeah, that does kind of look like a Christ Gorgia, actually. Can we get a zoom on it? Christ Gorgia is not a Sadie.
Not.
I think that we could probably collect that one. Although it's the first one that we've seen, actually, so maybe we should wait until we've seen other.
Yeah. So. So.
Oh wow.
Yeah, that does look like a black to me, but could we do just a brief zoom on the polyps? Yeah. Yeah, it's a black whale for sure.
It's a cool looking one.
It is, yeah.
Cool.
This looks like a lily pathies or something like that.
What's that?
Chrysopathy something?
Sorry?
We have one that looks like that, and it's called chrysopathies.
Okay.
Yeah. But with our drop camera, for the longest time, we thought it was an octocoral because it looked white,
Oh, yeah,
right?
yeah.
And then we go collect it, and it actually turns out it's like 2 or 3 species, and they're cryptic, and it's black. And at the exact same time, we figured out that Aristonella, which had a black skeleton, is a soft coral, and they were like, somebody should take these jobs away from us.
Adorable.
He looks like if a glass sponge with spicules and a fish had a love child. Oh no.
Oh no. Not again. What a cutie.
Yes. A little reflection there.
Yeah. I can see myself.
Aww. Yeah, that's the RV. Oh,
Yeah.
just like.
Oh, look at the little bobber. That's a regular fish.
And there's a wall right here.
Oh no! Why didn't anyone else say anything? You guys are monsters. Why didn't anyone say anything? Jeez.
There's quite a bit of alteration on that too. It's
That's what it
If we can get like 2 more in the area.
2 more what?
Corals, yeah.
I just passed over small thing.
Alright, next one we see.
But the next one.
is. Do you care what species it is? Just the location?
Just the location, yeah. Oh, there's another one on the left, but kind of hard. Sorry, I just saw it. It's okay.
Going or?
Keep
yeah,
going.
probably keep going. There's one coming up on this pillow here on the right.
That little branching one?
Yeah.
That's cool.
That one has a thick base.
It does, yeah.
We could put this in with the other one, maybe? Just... Oh yeah, that's...
And I also see some nodes, so this is also bamboo. So yeah, let's collect this one as well, please.
I'm gonna go back to
Still have a rough estimate as well.
Put it in the same
No,
well.
I have an absolute exact.
I don't want to screw with it because it took us a while to get it up and running
444 meters away.
the first time, and it turned into a disaster. So it's working now, so I'm putting a touch it. But for next dive, we'll load the new grid. I've got it right here, so I'm using it here.
To the right, you need a little more room to put it back from there.
Can you close whatever this is?
Yeah, I knew you were recognized it.
How's that? You want laser at the base?
Laser at the base, please, yes.
At the
Sponge?
the trunk.
Yeah. Yeah.
Beautiful. That's perfect.
Awesome. Yes, please. I think we're good.
Yeah, that looks great.
It's pretty good.
So we're gonna put this in the same box.
Same ladder too, or?
Yeah, battery 3A as well, if possible.
Yeah, might. You'll just leave in there
My only thought, Don, is if any of these branches break and that one breaks, then we're gonna have no idea which one's which.
You mean if they both break? Is there likelihood they'll both break?
They're pretty fragile.
You could, because they're the same species, right? You could choose to put them in different ones, and then as soon as you get a different species, then start doubling up.
Yeah.
Okay.
Is that okay?
Yep.
Okay.
So
3B.
same box, different plot.
Yes.
Alright.
Yeah, whatever other one, just not 3A.
Sorry to be misleading.
No comment.
BO2?
Yep.
BB3B. Now we are in the bottom of the valley now, as far as I can tell. So we're right on the axis.
Right on the axis. Nice.
You probably know this. It's just from Darwin. They're watching.
I did learn that in the first so I didn't put it in seawater right away, and I was like, nah. okay.
I should tell him early.
You
You isn't half the problem.
Yeah.
We're cooking now.
But thank you, no, I do appreciate yours. I'm reaching out.
Y'all swap the outer with the inner, right?
Yeah. My foot now. Yeah, that's what I said. Like 50 grand. But also that. 50 grand. But also that. I'll sign off.
You know, the black oil, just in the crack.
Correct.
We can continue on forward, or south, or we should go on here.
Yeah, we could start heading probably around 100. We'll head in that direction for about 500 meters.
Can it be up this wall or stay down?
Yeesh.
It's a small wall.
Yeah, I would just turn and whatever we get, we get.
New Waypoint Bridge.
0.4? 0.3? What do you want? For speed?
Oh, are we doing InSAS?
No, not right now, but we're planning.
Yeah, that sounds great. Thank you.
So that's an hour. 6 o'clock, 5 o'clock. Around 5 o'clock. And do we need waypoints?
I can eyeball them if you want.
Yeah, let's live on the edge. Is it? Yeah.
And I can give you that long.
Okay.
After we make it.
We'll have the backdrop.
Because we're not gonna know where we're starting from. So we'll just start from wherever we are, just set ourselves up here.
Yeah, it should
Do that. Is that right? Yeah. So 5 o'clock. So it's hour and 45 minutes.
Yeah. It can happen exactly.
So how many do you want? No more than 40 something and 45.
Can we put the camera up a little bit?
But some was and earning up to disappear for 15 to 30 minutes. So if you see it fall,
Can we pause there? Sorry. That bamboo sticking right out towards you. Can we possibly size that one?
What do you want to do with it?
Just size it and possibly collect it. Can we get the lights turned down if possible?
Here, if you look at like the 2 seconds,
Yeah.
the last second was right, and this one is shifted south.
Had you measured that offset?
No, Charles might not, but you could just see it, like,
Yeah.
but the offset from these two.
Okay.
So you're tweaking
It's like in the right range.
Okay.
It's about 10 centimeters because that laser is projecting further than it, and it's ahead of the lasers. So I think you're about that.
1687.
Yeah, sure.
Yeah.
I mean,
Okay.
if you
That's
say you're 436,
so that
you've
offset
seen
would
them pretty
be
consistently, and you want at least 3. Could you have more than 3?
30 to 40 meters, 35 meters or so.
Because then you got 3 in the bag.
All right, I have to program my brain to see that now.
If it's not, I think if it's not the right size, you could wait.
Okay. Okay, let's keep going.
We've been seeing that pretty frequently.
We have been, that's true. Alright.
What's the closest you would want to get to it? Because you still
To the vent.
Yeah. Because
I want to see what the heck that is down there.
But like, do you want a gradient or do you want
Yeah,
this?
I gotta get out of the way.
Hey, shells?
Yep. No, it's a fish, not shells. Quite different.
I'll see myself out.
We're gonna take a close look at detail.
Yeah.
So you see all these little white specks?
This is active.
Yeah, we saw those at Roseburg.
Yeah.
Yeah, those look like they might be alive down there.
Yeah. Look at that. Hey?
Yeah, it's pretty cool. Oh, maybe I'll take a highlight finger.
Yeah. Don't go through.
Yeah, let's get away from that thing. It's giving me creeps.
So what's up top?
Who wants to find out?
Wait, there's something right below us here. That looks like a chimney.
Oh, of course.
Where these guys are coming from?
Yeah. Let's take a quick look at this first, though.
It's got the same tube pattern to it.
Rock box. You want to have a look up before we start sampling this thing? Come back?
You want a sample of this, Mark?
No.
Okay. Let's yeah.
Judging from
Yep.
Karina's excitement.
Biology road.
Follow the yellow mussel road.
You go up to look. You can see it's all falling down.
What are our forward-looking sonar options?
Yeah.
this afternoon.
Yeah.
In general.
Oh.
Okay. Oh, there it is.
Ah, yeah. Yeah, look.
Oh.
My neck's getting sore. Neck's getting sore from working out.
Yeah, that seems bad for that. Yeah.
Huh?
I wonder if they were just living up on that cliff as they were feeding off the rising fluid.
I'm expecting something.
Huh.
Yeah, it's still super.
All right, let's go back down.
There's gonna be a event up there.
Oh, I don't know. I'm scared.
That's remarkably
So probably the venting was down below where the chimney was, and
Just enough to
Wafting up
with enough
and
coming
feeding, yeah.
Wow.
Old burns?
That's what we used to be. Some fluid.
Yes, sir.
Here.
Okay.
Oh. Oh, our positioning is now
But not that long ago.
right.
A few years.
I think we're there. What's that? I think it's positioned correctly. So position is good. I think so, yeah.
This is only a few years ago.
Awesome.
There's just some filtration.
Thank you.
Yeah, so that's why I'm kind of debating if I should go back and get that small one now.
Do you want something close?
Very close,
Yeah.
Photo, are we, got it?
Yeah, John,
Yes.
would it be possible to go back to the coral that was just right over there? Because it was really close to what used to be active venting.
This ROV's got an ambi turner. Have you seen Zoolander?
Oh, yes.
Have you not seen Zoolander?
Then you don't get a quote.
Sorry.
Anyway, yes, that's a great idea. We'll get the coral.
Okay, thank you.
As long as you, right after this, go watch Zoolander.
Noted. Will do.
This one right here? No, that's a fish, never mind.
Ah.
I was hoping no one noticed that one. Was it at the bottom or was it on the wall?
On the wall.
Right down there.
Oh, I see it.
Yeah.
We've got quite the list going. We're gonna have to like dedicate some time to this.
It's an effort.
A really long list.
Zoolander's one of my favorite
Really watch
movies.
We're approaching the Seafloor. I have a feeling that we won't quite get to.
Did anybody mark or note that recently active site?
I did, not with a marker, but with the observations.
Perfect. Yeah, thanks.
Which one is that? What number?
For bio?
Yeah, is it on the bio thing?
We're at BO3.
Oh, it will be 3. We've plugged it 2.
sorry, I don't understand your question, John.
It's okay. B03. Were you referring to one of the markers on the?
I just asked if Morocco had marked the one we just found.
Oh, I see.
Was that for laser placement?
Yeah, that looks great. That looks great.
What do you want?
If it's small enough, can we, and you get the base off, can we try to suction this guy? Where it's so small?
Yeah, if for whatever reason you don't want to do that, you want to put it into a close cover or use it. Just so I know if I need to cover off or
Biobox.
Biobox, okay.
Yes.
What, J-Rod, do you think it'd be worth it to try putting the suction on top of it and then scraping the base?
Between all of them. See this one?
I don't know.
That
They look all.
I think you have a very likely chance of snapping it while trying to just line it up.
Okay.
I mean, we can definitely try, but.
Well, whatever you think, it was just a thought. Yeah.
Is that the way you want us to try?
Why not just do it the regular way?
Okay. Yeah.
Are we tight on coral quivers for this dive?
I don't know.
The bioboxes are assigned to corals, and the coral quivers are assigned to sulfites, and the front biobox is assigned to eggs.
With my eggs, if we continue to collect corals, we could always put a coral in with the eggs.
Yeah. Okay.
Yeah.
Cool. Thank you. And we're not recovering the ROV at 6?
No. Nice.
That's great.
you want to put it in
Suction sampler.
I'm lined up on one.
Suction sampler 1. B03.
It's quite large.
It is quite large. J-Rod, do you think that's going to get stuck?
Very likely.
Okay, let's just not do that, because then we lose suction for the rest of the dive, and that's not worth it, in my opinion. John?
Yeah, that's fine. Biobox.
Biobox.
Is it the same as the other two, so put it in a different pot, or is it
Yes,
double?
please. Yeah.
Different pot, alright.
It's just like, especially with that bend, I just see that immediately getting stuck.
And biobox 3 is coming.
Oh no. Just make it run for it.
Face.
Come on, any record of volcanic limestone sample here? Any record?
Did he just swim in?
Yeah.
What?
I guess not. I want to shrink. What
I
happened?
don't think so. I want to pick up a.
You're on biobox 4.
Yep.
So 3D is where that ended up going into.
Maybe 3D.
It's coming out.
Yeah.
For the data loggers, it's Biobox 3D.
Don't close the biobox yet.
Yeah, he's coming out.
Can we put a rock in with the coral? No?
Unless there's a way that you can make sure that it doesn't squish and break it.
No, not at all. Okay. We're gonna pick up a piece of basalt.
From this area or from
Yeah,
the
just
claims work?
just one right in front of us. There's one sitting there. But that's not gonna go in the bio box. So if you want to worry about
it's
closing
not
that,
going in?
yeah.
Alright, so like it. keep up.
Not moved very much.
Okay, so okay, yeah, okay. So
Who's your name? Steve. Now it's gonna go into what, a quiver?
No, this will go in rock box. The rock box one.
Like a nice round one that's sitting there all proud by itself right there in the middle, or you
Whatever you think best suits the mood.
think
Don't you wait me down.
that one is
Yeah, there you go. I
No.
like it.
You're getting off in TRs anyway. What do you care?
No, just, I think you can reach it, right?
Maybe.
that's really great. Thank you for coming back for your decorum. Appreciate it.
Where you're a little bigger than that.
Perfect size. Is that good? All right.
Good.
Marcus got a note from Z. Said if you're reading this in real time, there was a large radial fracture pillow where you saw the clams just before sampling the coral. I suspect it is at least basaltic andesite, if not more silicious.
Yeah, that's where we're taking a shot.
Yeah, okay.
Is that like the Z Nina?
Yeah.
Rob Zimber? Yes.
Oh, he's retired. Why is he not?
He doesn't retire.
From the pre.
He can probably hear us.
Hey, he's watching.
So you're not gonna be
Yes,
shit
buddy.
corals anyway? Yeah. Just 'cause we weren't at here. Makes we weren't at here. Yeah.
So this is RO1,
Yeah. Yes.
RB1.
Yeah.
And then
Do the coral first.
no in there.
No, and then.
Yeah, unless you trip and fall over for 15 minutes. No, you're good.
All right, let's head pretty much due east.
Great job. You can carry on that loop now, please. And by that move, I mean 7 meters until we see something else. I can go back to my room with these. Come on.
translate down slowly to the left. To the
There's an egg. Sorry, it just flew over one to the right. I'm not suggesting we pick it up, but can we just tilt down and take a look at it to confirm it's an egg?
sure.
It's in the hole. I literally just said that. Right there.
Good spot.
How did you see that?
It's rocks and eggs. I have a space. That's it. Oh, that's cool. Okay. And it does look like it's broken open. Okay. Thank you, that's great. Can we make a note? We got an egg. Our first egg.
Egg case.
Egg case.
Specific.
Well, it is.
The eggs inside.
The eggs were inside.
The eggs are inside. So we should have
the story of this egg nursery observation. So, later.
You should install like a secret manipulator arm underneath. Control and just sort of grab that.
What's that?
I spelled it wrong.
You spelled the word egg wrong?
It's 2 G's. 2 G's. Every time.
It's a really yes. So in this area is where all the egg cases are.
A ton of geochemistry here, John.
There's a ton? Who? How old?
2016.
Oh.
So you thought the rest of the box was sulfide.
No, it's okay. It's okay.
I had literally just said the other team sold them 500 meters away from the fence. And then I'm like, and look, they moved it. It's like it was clockwork.
Oh, I like this. Peek over the edge, but just keep moving in a general direction. What's this in front of us?
The layer flows. It's cool.
Right, Darby. Okay. I don't know how quick it was.
Big thing on that rock.
Black coral?
Yeah.
This is, you know, Here, is that one up there? No. No, it's a broken whistle.
Okay. Trickery with the shape though.
Yeah.
I'll zoom in on something. Or it has to do a spinner.
Yes, yes, yes. I'm always seeing things too.
Nothing real green up.
No.
Oh, wow.
Were you in here when we just sold the first skate egg?
No?
Yeah. It was broken open. Skate egg case.
We saw it. She saw it.
She saw it.
You know, we keep them busy and not moving forward. That's okay. It's like 3, 4.
Probably another
So
300 meters or so.
430, 440 meters away from it.
It's beautiful.
Yeah, we could do that to kill time. I mean,
But I would wait until we get to the event to do collections.
Right, Jeremy, I've been quite right.
But determining if there's viable ones at this far or if they're all just broken open. I'd love to know how long they persist for.
Saw kind of had some tears.
Yeah, so that's
some discolored.
Yeah.
How do they hatch? Is it through the egg? Or
Yeah.
do they come out of the egg?
If they hatch, it comes out the end.
Yeah.
If it's predated on the middle, will be torn open. Yeah. Yeah. And then they also get a lot of biofouling on them.
Yeah.
Afterwards, it doesn't necessarily mean it could be in its later stages, but biofouling and a tear is done. We did get some rocks too.
Lights aren't really quite right for it. Yeah, you're right. Those are previously mine sites, the numbers.
yes.
Where's your map that you made with all the closer to one?
So let's say the red circle. So if I zoom out. They are not. But over in this area, there aren't very many.
Just want to give it a heads up in like an hour and 10 minutes. The dial will start.
That's when the burn is?
Yeah.
Hour and 10 minutes. So 450, okay. An hour?
Yeah. I think a little less, actually, like 45 minutes. A little faster on the way up, so yeah, I think. So, I think it's 5:30, 5:39. It's like 5:40 or whatever is the expected surface time.
Okay.
Based on the descent, or assembly last time.
540, so we have about another hour, and then we're gonna do a transit. So we're not actually gonna recover. I don't know if
Okay.
you're aware of this.
I wasn't sure, so that's why I was kind of going to tell you.
Yeah. So we are, we're just gonna drive over there.
Okay.
And
RV will just be on bottom.
RV will stay on bottom, and the ship we'll figure it out.
Okay. We'll be able to track it at least. So.
Can we track it now?
I can ask. Yeah, see if they can ping it. How far away are we?
We are. We're 1.8 kilometers.
Okay. Yes.
Yes.
Oh, I didn't see you over there.
Okay. Yeah, I'll go. Let's go ping now.
Yeah, might as well.
Yeah.
And then on the ascent, I'm guessing it'll continue to drift in the same direction.
Yeah, further east a bit.
I don't know. I didn't know.
Hey, maybe we should make that assumption. But
I looked at the ADCP earlier and it was still east.
yeah,
so,
okay.
Yeah.
Anyway, what the plan is so there's a bunch of venting in here. We're over here just working our way towards that.
Yeah.
In an hour. Less than an hour. We're gonna run a SAS line over to here, and that'll take just under an hour.
Okay.
Theoretically, that time you should work out,
Perfect. Yeah.
and then we go get it, and we'll know exactly where it is because the USBL, the iridium,
Yeah. The radio.
and your radio,
Yeah. And it'll be a little darker, so we might.
and
see the
we'll be able
strobe.
to see the strobe.
Yeah. So.
there's no way we can screw this up.
Famous
Totally
last
sorry.
year.
You just jinxed it.
Remember what happened when you said? That wasn't even that long.
I'm a slow learner.
Alright.
Okay.
I'll go pay it.
Okay. Tell me again the release time.
4:52.
4:52.
Yeah.
And 45 minutes.
Yep.
So 5:35.
Yeah.
So 4:35 is when we have to go. I better write that down. 4:35, which is in. All right, that's good. Okay.
Cool.
Good. Thanks.
That's our current meter descent. We just literally landed right there.
Oh no, yeah, I saw that. Yeah, I was like, holy.
They started to manage us and I want to enter and wanted us to land like 100 meters away and then come up to it. And that's supposed to land on time.
All right, we essentially want to stay around here. If you're gonna go in any direction, veer right instead of left.
You're right instead of that. Yeah, just been zigzagging to the bridge and going back and forth. That's one.
Yeah.
Come to this bridge and then turn back around.
Yeah. Peek over the side into the abyss.
Would it be possible to get an estimation of where the ROV is relative to the active point right now?
So which point?
Like the active vent that we're going to be working towards. Yeah. Yeah.
What do you want to know?
Just distance.
Oh.
I thought the active stuff was down in the deep stuff.
Yes and no. But yes.
Okay. John, would you be able to?
How far away are we? Yeah, I could tell you. And 140 meters.
Okay. So we're gonna hopefully try to collect a couple more if that's okay.
Yes. So the close floor.
Photo. So that means about 10 meters down at the top. The top is here.
Yeah.
Not worried about that, right?
Nope.
John, do we have a new ETA for deck time?
Yeah, no, is sort of the answer.
Should we go more south than this one, or about there?
No, this is good.
Okay, so east from here.
Yeah, east from here.
Still zigzagging in and out here.
Yeah, the depth of the vent, as far as I can tell, is 1687. So it's deeper than us.
Or maybe off the side?
Yeah, I just wanted to see if we want to see what these things are over here, but I'm not sure I want to do that yet. Yeah, I do, actually. Let's, we just head south?
Head south.
Just for, like, 30 meters.
Should we see the remote?
Wall.
There is a wall. That is correct. Very big wall.
Is Slater House updating?
Seems like it's either stuck or behind.
Let's see.
They're moving a lot for not moving at all.
Reasonably recent, but not going sound right down there.
Yeah. We
found it.
here it is.
Flutter mouse is updating now.
Oh wow.
It's just
Okay.
cruising.
There we go. Alright.
There's 10 meters.
So you want me to go along this or back
Now
down?
let's go back down.
It's interesting to me is the number of black corals.
There's some features on the map there.
Certainly compared to where we were.
Yeah, and how it's shifted, and you can't really. Well, we're not seeing as many either. There's just smaller.
This thing here.
Are we going back here?
Yeah, somebody jump back off the cliff.
He's trying to zigzag.
Oh, I see, I see. Okay.
Or are we up in Waypoint?
So we should start seeing.
You swing right a little bit.
Right
See
on
that,
this.
that chimney up there? You wanna look at that chimney?
Well, there's alteration.
Yeah.
And there's a chimney right here. Chimney there.
Chimney or wall?
That's good.
And this is all.
How about that?
Aha, it was a feature.
See some microbial matting down below and then between the.
Yeah, there's some venting here.
Yeah.
There is, yeah. There you see all the muscles back there.
Egg case,
Eggs.
just below the median. If you just tilt down a little bit, then 1/3 where? Nope. Okay, belay that.
Oh. I even got my poker out.
Yeah. It's basalt that was masquerading as a egg
At least we
case.
know you're prepared, John.
John, would I be able to get a rock?
Yeah, sure.
A lot of that is just loosely cemented.
You want all the gooey stuff on it? You want a rock with gooey stuff?
Yeah.
Oh, yeah, we'll have
Oh wow, so many.
Oh wow.
Oh, moly, that's impressive.
I love these guys. Cool.
All of a sudden it's wow.
So those are amphipods, logger.
Yeah.
And there's the
And so-
tepia in there.
Yeah, I was gonna say.
Right in the middle, do you see the little red plumes? So that's the other species of tube worm.
Oh, yeah.
It's a tiny, very small, right in the middle of the patch.
Yeah.
And the big crab.
Little
our well.
Fish cruise now too.
Come.
These are mussels.
Nice.
Beautiful.
What makes them different, Lena?
Well, it's just a different species. It'll never grow as big as Riftia. It's more like it's quite closely related to the tube worms and Wandafuga.
You see they brought the multiple ruffles that makes the tube.
Cheapers, look at those amphipods.
Only the eggs, if you find them. But,
I've never seen them. I'm just wondering what these small fish are.
I'll be back.
Gusanos,
Very
gusanos calcáreos. Se llama
Possibly be the.
Let's let them start. Yeah. I think they're pretty kitty back. Are we highlighting this video?
Definitely have different ideas of what we
Vloggers, are we highlighting this video?
Yes. Even a lot of it. It's just.
Se convierten en energía química, en materia biológica, y luego se convierten en proteínas.
I remember reading the paper about the powder.
Interesting adaptations not seen. It's
Is there anything else anyone wants to zoom in on here? Going once, going twice. Okay.
John, I think that
That's what we're gonna get right now.
Where's it gonna be going?
Twitter.
To the right.
We're going to the furry rocks.
I thought we could get the mouth parts.
We know you're the expert
No,
now.
like-
Identify species based on the mouth parts.
Any Twitter is good.
or whatever.
Oh, there's the first maxilla, the second maxilla, the third maxilla. First maxilla pad. Mandible and then you.
Sí, yo sé, pero es que también toma tiempo de investigación, yo que sé, soy un tiempo, entonces escribí un mail ahora mismo a preguntar si podemos conectar
Moronke, do you want a temperature here as well? Is that? Yeah. Alright, so rock first and then, and Niskin 5 liter.
So maybe Niskin first, and then rock, and then temperature?
Oh, okay.
And lower rock, upper, I think it has to be upper. The lower stuff is all cemented, right?
You want some of this stuff?
That's right.
So this stuff right here?
But you want a Niskin first. Okay.
Right there, either on that thing sticking up.
Yeah, he wants to spire.
Okay.
5 liter Niskin?
5 liter, bottle 1.
Marina, what are the white patches back there? You see the white patches on the,
Yeah, I thought this one was, but
are they sponges?
that's what I thought.
I think so. We were just asking.
Some just like us. Should they like that?
Yeah, they can.
Thanks, Gerard.
I can ask for it.
Yeah.
You tell me what you love, and then I ask.
Good afternoon. I don't love anything.
Thank you.
I would love to publish something. But my question is,
You know what you should do.
All right, we got Claude and Chris jumping on the controls here.
Just bring
yeah, please do a simple drawing with the black person of the content. Please do a simple drawing with the black person.
How did that work?
It's like hard to accelerate.
Number 1. Right, Niskin bottle 1
Yeah.
for Niskin 1.
Yeah.
Did the loggers get that? I'm not getting any readback. Thank you. Next is a rock. There's a little spire on the big chimney. I think that guy right there.
In this size, in young little juvenile, we knew they were here.
And we haven't collected it.
But Ross ain't collected that. No, we haven't.
It's gonna go in a quiver of your choice.
No, no, they'll get like this.
The tube is much more rigid.
So they're different. They don't need as much sulfide as Riftia does to support its huge body. So they're a smaller body one. As you can see, where here Riftia isn't. Do you know what this is though? Yes, but who?
So, depending if you ask me.
So that's not recorded. But that's.
But that's basically the responses.
We know a lot of stuff people. Yeah.
Anywhere in here, or is this
It's a bit higher up.
Higher up.
Yeah, any of these things.
We could press on to this.
Yeah, I think so.
Do you know the collections?
Only one way to find out.
John, do you see a bunch of really small dots on the side there?
Oh, all this stuff?
A little bit up, right there. Oh, top, down, there.
Oh, there.
Yeah. Can you get a zoom on just that patch really quick?
Yeah.
If they're okay with letting you have the joystick there? We just don't really know what it is. It could be like hydroids maybe, or like solitary tunicates. Or
Maybe the zoanthid again?
yeah.
The small one?
Yeah, true, the zoanthid probably. Yeah.
It's weird that we hear them in the small patch.
Yes, it is. So there's a couple species that could really hear them. Including one that was described only from well, actually, the larval forms in the there's a later node. Standing
You wanna look at the little ones?
Yeah, just take a zoom in on that stuff over there.
Oh yeah. So the same zoanthids, yeah. So interesting. Okay, thank you. What is one? There's zoanthids. Z-O-A-N.
Yeah, and it's coming, it's growing because of the influence from the flow below. It's not growing because of the rock. It's growing because the fluids are coming out.
That hanging out. Is that?
Well, that's a good question. As in this too.
Oh my gosh, it is! Ah! Okay, this is a hydroid. I think of candelabrum.
Oh.
Oh yeah. Totally.
Good for you.
Yeah.
They're so cool. I got one under a microscope on the last cruise. I think it's the same, like, general species or sorry, family or group or something like this. But this is a different one. But they're amazing. They're amazing. And I don't think you can record it down here. I could be
No,
wrong.
well, not associated with events. These are generally.
That's so exciting.
And there was another one over here too.
Oh my gosh.
Could be.
I don't think so. But they were found around bends in the mid-Atlantic, which is super cool.
Yeah, whatever you can grab. But one of the furrier ones is better, so on this side is better than that side.
Okay. Alright. So we'll see what he did. How about
I would want to take it.
No, probably not. I'm so excited. Are you vlogging? Just like, sorry, I just got this. No, I'm not. It was an excitement. It was an excitement vlog. Yeah. Sorry. So neat. I'm just impressed myself.
No. I think candelabrum.
You know, I was trying to go south on. There you go. Good for you.
So, Verena, the ones that we sampled in the mid-Atlantic look like this. Yeah. Yeah.
It's 13. Wow, I wonder what its distribution is.
This is exciting.
Yes. They are.
So,
that's the
it's
other
slightly
species.
different. I
That's
think
the
it's a
next
different species or group or something.
Oh,
like this.
sorry.
So we wanted to show this.
Is this
So John,
Oh,
the one with the salt loss trying to be okay? Or would you rather one further to the left?
No, that one's fine.
Whatever you're.
Yeah, that's fine. Plenty of goo on that one.
It's the hydrothermal fauna, which I have. I've got all this on my computer, so yes, I can send it to you.
Okay, so it is associated with fence.
I guess so.
Is there venting coming out?
There you go.
behind?
So this was, that's described for the specific ones. So we can add that to our list. Isn't it?
It's occurring both in Atlantic and Pacific Ocean.
Okay, so it's a generalized, supervised, yeah, okay. I guess we'll look at the date on it, 1780.
You know what? This one behind might be just as easy, and I see fluff on that one, so.
I didn't realize because on
Okay. Just make sure I get it low enough to get that little buzz. Did?
Yeah.
Alright.
It's all right. It's a senior moment, my dear.
Obis is what I'm thinking of. The distribution doesn't seem to. Here, I'm gonna look it up. I'm gonna look it up.
Circulate, that's one of the two circulate species here.
One.
How much fuzz I got on?
This will be alright.
Big one.
Okay, so this makes sense. The one that we found on the middle, I think, has only been recorded. The one.
It seems to be known, like certain, there's a name for snake pit.
Right.
But the other one, Phrygium, PH. Yeah.
But still, I don't see anything with the surface. At least on this.
Oh, this. Yeah, sorry, I don't know. I have so many errors and I never use it. Well, it'll tell you something like this, but then I know of a lot of errors. A whole lot of stuff from my own collections that is in there, and I never sent it, somebody else. Yeah. So. Anyway, it's not much. There's quite a lot of them, aren't there?
It's just sitting on there. I can't really make it down.
Cool. Just a little bit more mostly on that bottom area. Because there's quite a lot of those Android and
Okay.
Galapagos. So
Just in
get
that
the
area?
No, just the whole thing. 'Cause there's about five of them, 4 of them down here. So
Oh, I
just to
see,
be
I
able to
see, I see.
That in
Yeah.
the image would be really good.
Okay. Got it. It's just sitting there. It's
Oh, is that trying to eat?
Oh, yes. Oh, dear.
That's running away.
There goes one of these.
nice.
This is RO2, Coral Quiver 7. No.
Need the top back on it. 8?
Yeah.
Oh, sorry, that's what you're doing. Okay. Yeah, it's down the other side.
Yeah.
That's why.
No.
I don't really care about it. So you can get it next time. So you're gonna do that, right? I'll give it a shot real quick. You might need a layer. Your headphones, I don't want to get away with it.
Real benefit is bridge.
It was named for the site.
This is circumarctic, but it shouldn't come to south. And there's also some taxonomic problems with the name.
So it's probably not.
It's not that.
Yeah,
It's describing what.
That's fine.
Causing more damage to the rail than
Well,
top is worth.
there's a specimen right there. Yeah. You want to describe it? Do you want it?
Yes.
You asked?
Yes.
He's looking at it. I think we agree it's probably not. It's probably not the name that anyway. It's in the handbook as something for this area, but
Just take the camera.
it just doesn't make sense.
Marina,
Yes?
you want to take a close look, or a closer look at? Is it in here that you're looking?
Yeah.
Okay, so what's good about this is just getting the image of how many are here.
Okay.
And what we're sitting back here debating is whether Belen is going to ask you for a collection.
Oh yes. I will. If you don't mind.
Because we think it's a
Yes,
new
it's still
record.
hydro.
And probably an undescribed.
So is this a suction sampler, sort of?
Or is it a grab on the rock? I don't know which would survive best. See the little dude, the stringy dude? All these guys, whoops, and this one. So I'm just wondering, is it gonna survive better by suctioning or by taking a piece of rock?
That rock is pretty complicated.
Yeah, my feeling is suction.
So you've got a few you can try, whatever comes. There's quite a few there. So it's an Iberian, sort of related to corals, but and it would go to the foundation. Hopefully they could get someone to work on it.
Ah, okay.
So no, I'm not. No, it's not.
You're going to make this very complicated for
No,
all
no,
of
no,
us.
no, I just asked Belen, and then she nodded. She said yes. Now, there is a species described, the one that Monica saw at the Mid-Atlantic Bridge, and it's described in the hot vents there, that is endemic to the vents. So it's not unlikely that this is something novel for this area.
Where we saw it on the Mid-Atlantic, it was not directly on it, but a little bit beside.
It's sort of is a little bit beside it. It's peripheral, is what I would call it.
graphite, it would be.
That
when
kind
they,
of good.
when they come up, it'll be like very little structure. Yeah. Like it'll just be a bunch of.
Yeah.
What do you need to decide? What's that? What do you need to decide?
There's one from our season in the back. Just swam through.
Now,
So they're there.
Oh.
Oh. Still got a job, so. Not a big deal. Watch it. I got it. Bring in the better grip.
When do we need to be on site? John? When do we need to be on site?
we're gonna have to leave in a few minutes. We're so close to the high temperature bit.
T-probe.
And the disease venting site where all the muscles were?
Yeah.
You don't want to take it from right here?
Is it, I don't know if there's venting happening here.
Well, this is where the rocks come from, right?
It might not need to
Check here first.
Yeah, I think so.
we might still be able to breathe.
We could drop down and take another one at the bottom just to see what the maximum we get.
Okay, so that works for me.
Okay. We'll take. Where's my thingy? We'll just get a temperature right where all this bacteria is, just to see. It's probably not going to be much, but.
Yes, of course.
Right. It's about time to start moving in that direction. Are you guys talking to it?
I was tracking it for this year.
Yeah. Is it moving? Or it actually hasn't?
It's not yet. Sitting on that.
Okay.
It's also unknown when it will start to descend. I guess there's a thing that's occurred on the road. But last time it was like 8 hours.
Yeah.
This time it'll be 8 hours. I don't know.
Let's see how we're late.
What was your plan?
Yeah, just sort of stuff it in here somewhere. What's going on with that arm?
That way from this.
Pardon me?
But bring the way from up there. I'll be the
2.78.
278. Okay, you get that?
Yeah.
Yep. Alright, and you can just leave the T-probe in your hand and we'll just drop to the bottom. Stuff it in down there.
Alright.
I'll just hang it down in there. Actually, yeah.
Oh, right. That's probably a storm reducer.
Oh yeah, of course.
That's what it is. That's bigger than
Because we've seen this big collecting along the Fugal Bridge. It was never described. 2 specimens with samples
At this size. Must be.
Yeah.
Okay, trying to get
Did you see that earlier storm you see over off to the side?
A little 3?
Storm Medusa. It's they're stuck to the bottom and they have to like a
On the
little
left
cup
or right?
with little
It doesn't matter.
earmuffs all the way around.
Oh, I don't know if I ever seen one.
Alright, there's nothing else there. Okay.
Anywhere else?
No, I don't see any.
Shimmering water at the base of the chimney.
I don't know where that is though. I know where the base is, but. Can you zoom out?
Oh, yeah.
Is it?
Yeah.
Pan right?
I don't know. Don't go with that.
Tears of Hope and Terrace on this almost.
Alright, let's take off if we see an obvious spot.
Alright, we're gonna hold it just in case.
Yeah.
Yeah, they're missing.
Oh, yeah. Wow.
Right, isn't that sweet?
Just picture
You said Soma?
Storo.
Storo.
Medusa.
I think I write that down.
Storo. Stauro. Medusa. Biodiverse. That's good.
That's good.
No.
We're gonna head around 050.
What about down there on the right? Is that where it's venting?
That's what I was thinking.
Oh, down there.
Lava?
Do you want to grab the
Also
temperature
on the
there?
right.
I would like to try it.
So let's go down to the left.
He's going left. She meant right.
Right.
You don't want to go where all this.
That's where the diffuse vent is probably.
Yeah.
Yeah.
That's
so
not what you want?
I do want that, but if there's actual venting coming from the where we collected itself, so I'd like a measurement there.
Alright, well let's grab the temperature here.
Yeah, those
Jump over there.
Alright, so in the middle of the.
Yeah, just right through there.
These little shrimp, Verena, what are they living on in the water? Aren't they normally grazing?
Oh, the little amphipods?
Amphipods.
Yeah, they seem to be eating them, collecting the bacteria that are coming up.
Right out of the water.
They have a whole bunch of little mouth pumps that sticky, seedy, they pick up.
They look like mosquitoes in yellow.
Yeah, there's something we never see on the field, that group of critters just never appeared there.
No, I didn't have that view in the lab. You're welcome to Deep City.
You got your phone on you.
Oh, that comes off. Is there a hard stop for starting InSAS?
What was the
5 minutes ago.
Nice. Yeah, we have to cover some distance to recover the land, and so to go over. So I think that's the lens to SAS surgery, where the land is. That's a good thing.
Yes.
Remember when they said it might go faster than we thought? I think we jinxed
0.4
it.
instead of 0.3.
I think we jinxed it.
Makes a difference.
And that should be happening now.
396, 410.
425. I can go a little deeper if you like.
Yeah, sure. Might as well.
Swamps anywhere else, is it the depth?
That's different rocks up there.
Yeah.
They got cooler. Yeah.
It's interesting because we didn't really see this here. I'm just thinking that maybe there's just not enough flow. I don't know, because they.
498, 493.
It's very good, isn't it? Yeah, I don't know. It's like, yeah.
Yeah, we can go with 527.
Alright.
All right, done. Farina, Rob is asking, are there any molecular clocks that suggest when Regia separated from Oasia? Oasia. Oasis.
Oasis. Yes is the answer, but do I know when? The work was done by Bob's son.
Where are you seeing this one? You want to check out?
Right in front.
Under the chimney. So where it's nice and white there?
Right there.
So you guys got very distracted. So fake eggs?
Fake eggs?
Oh, yeah, tricky. At one point, you guys had gone back to the original cliff, the little sulfur spire, but this is a new one, right? Yeah.
There's a little shimmering down there.
I'll see. Must the left. Bring your we have the push course lever to get in the rock. Push course. This will be just gonna bring that rail in or you can set it on. Might be easier to bring that in some other way.
322?
322.
Happy with that?
Yep, I'm happy with that. Thank you.
Yeah. Go ahead, Rich. We're coming up right now, so you can head that
Sean.
way. 600 meters due east.
John, could you unplug this?
Oh, 600. Okay.
He didn't trust me to
And
do
we're,
that. Yeah.
we're not gonna go due east. We're gonna follow the ridge. So it'll be, I don't know, 100 or 110.
Alright.
We're gonna try to within a SAS along the way.
Alright. Where's your ridge?
So we're essentially going to drive in a straight line following this ridge until we get to here, and then just jump up. And that is where the lander.
Yeah, it looks like we're pinging it.
Okay.
We're gonna try to do this at 0.4.
Alright, so we're done. I was doing that, so.
Oh, I should have. So we're here, we're gonna essentially follow this ridge. Go
Okay.
back down to here and then jump up.
So do you want a 3 flat or do you want to use the other one?
Probably Caroline on a 3 flat, or if we put a
Waypoint is easier because it's more stable. You know,
Right.
to apply that.
yeah.
Okay. Alright.
I could give you a position.
Yeah, that'd be great.
Here. You want to go all the way?
I want to go all the way to here because then we catch this stuff.
Yeah, I'm not sure if this is high.
So we could.
Let me know. Do this and then change.
I don't want to complicate this.
Okay. Then let's go here.
Here?
Yeah.
Okay. Just punch in coordinates, is that the easiest way?
Hold on one second. Alright.
Is it decimal degrees or degrees minutes?
Decimal degrees.
Decimal degree.
Decimal degrees. Okay, so let me get the spot here. All right. Longitude 2.1026.
2.1026.
Correct, north, and 91.9252 west.
91.925.
Correct.
Alright, you want to go 0.4?
0.4.
Alright, and an altitude of
15?
meters.
Alright, so you said the 15.
That might be a bit of a.
Alright.
Are you using a snap for this or
No.
Okay.
Alright.
And the idea will keep the ship in front of us.
Yes. So the ship is pulling us. That's what works best.
You might have to have your altitude up on your own.
We need like a minute just to start the SAS.
Alright. Yeah.
Should we start the line from on top? Or do you want to start?
Waypoint is disappeared. What was it again? 2 point.
Yep. 1026.
Current folder to be folder for storage. I can also click on the data based on the hospital. It should just
What?
91.9252.
Would we be able to see anything in the sonar at this height? Forward-looking sonar?
Possibly, because we're going to be going over some stuff.
Probably an idea to have
The lander's coming out.
We're on our way.
Okay.
Ish.
They just be about the same thing. Same numbers.
Have we started?
2.6. Yeah, same zones. So 208. 5.
Oh, wait.
Start with the system.
Yeah, it's 5. Alright.
208.
And what you needed?
Sorry, 5. Yes.
0.4, 0.3.
0.0.
0.4.
Alright, go up one.
Yeah. That's why I'm not on WhatsApp.
If you want to change the heading, you want the heading to do something different than what is on the waypoint stuff. Yeah, if you want to change it, you have to grab it. If not, it'll just do whatever it is on when you set the waypoint for the rest of this. Yeah. Yeah, you should be going. Bridge RV to that waypoint at about 0.7. Thank you. Alright, so what's that again? Wait, no, we want to go for the full shit. I forgot. Sorry. Very rough.
Do you want to close?
Are we started?
Yeah, we're going.
We're going?
Oh.
No, we're good. We're good.
We're thinking.
What do we want to call this? 912.
It is 23 overall.
All right, so if you've been able to hear a little bit in the background, we are back up about 15 meters off the bottom. We're going to run a little InSAS survey on our way towards where the hydrophone lander is coming up. Oh, there's some smoke over there for sure.
Yeah, we're going right by the chimney.
Yep. So the lander currently coming up, it's down about 1,480 meters or so, and it's on its way up. So we're basically gonna survey along a ridge here on our way towards it, and it'll actually bring us up a little bit south of where the lander's currently coming up, and then we'll just scoot north and find it.
Can you make a note of that smoke?
So it does have some beacons on it. It's got radio beacons that we can use to kind of sweep for it, but then it's also got some flashing beacons on it. And as we are approaching sunset here, those will become more useful. So we are on our way. Again, this lander we're going for, this is the hydrophone lander. It got left behind near the site of some black smokers. And we just kind of scooted off. During the night we went further away, but where we were working just now it likely heard us. But overnight it had some quiet time to itself, and hopefully picked up some cool recordings from the venting areas there. Originally we had planned to recover the ROV and then go get the lander and then put the ROV back in the water, but current plan is to simply just fly over that way with the ROV in the water, and then fetch the lander and just leave the ROV down. So.
What's that? Come back, I didn't hear you.
Yeah, the way you put it on the wing season, that seems to be the site west, or further on the road on the bottom.
Correct. They want to map this run until we get to the end of our waypoint, and then we're going to turn north.
Okay, because the landlord should get to the
Roger that. So, the expectations that the
Lander is just passing 1,400 meters now from the surface. I mean, do we know how long it's gonna? Do you know how long it's going to take us to get there, to the end of this waypoint?
It's 1,500 meters.
We're going.
How fast are they?
We're doing 0.4, yeah. Oh, the A snaps. That is we can set that basically to take a picture in the log, or take a snapshot in the log. Every 10 seconds is what we usually run it at, but when we do the photogrammetry, we actually set that to every second, so that it's just catching a picture every second.
It's about the same time that we take the land.
Yeah, like it starts out going this way.
Yeah.
And it's going to go that way.
So we're currently discussing where the lander is likely to come up and how it's likely to drift versus where we are headed. We are deliberately headed a bit to the southwest of where the lander is currently, but we think the lander may end up drifting north. So it'll be even further away from us, but we're going to see. We've got about 50 minutes or so to figure it out. Have one. And we are able to track the lander as it comes up. It has a beacon on it that we're able to ping. It's the same system that we use to actually track the location of the ROV and the CTDs when we have the CTDs in the water.
This drifting slowly east.
East, but not too much north.
Right.
Okay, that's all right then.
Yeah, I don't see any.
Well, that's very slow.
Yeah.
I don't see any currents of in the north. Means I don't know what the hell's
I don't know, we'll see.
Yeah, I trust them.
We are looking at pretty calm weather out there. There you can see the view past our heli deck. So current ETA on the lander reaching the top, reaching the surface, is about 35 minutes or so from now.
Yes, I'm trying to figure out. Oh, I see. Do you really are? Yeah. You mean, I got it. Alex. Get us on the screen and Golden Mercy.
Which one? The screen?
Yeah, I just wanted to know that.
Oh, hold on. There you are.
Step back. Rocks.
Carolyn, do you have a suggestion of how we could cut it shorter?
Something's just like that's
We need to be, we need to be up there in 50 minutes, not at midnight, fallible, not at 59. So we're gonna have to do something differently than the transit. Still, we've got time to ask John, and he wanted to get the smokers.
Yeah.
Yeah. It's over here. Right to the left of the group. Thank you.
we are currently on our way to where the hydrophone lander is coming up. So the hydrophone lander is less than 900 meters from the surface now. On our way over there, we're doing a little survey with the InSAS, the synthetic aperture sonar that we've got. But we're gonna scoop this lander out of the water and then head back down and do a little more exploring. It is almost supper time here on the ship, so you'll hear folks shuffling around for that probably as we come in and out. But yeah, for folks just joining us, that's what's going on. Is we are just scooting about 15 meters off the bottom, give or take, as we head towards where a lander is headed back up. We'll scoop that out, and then we'll touch back down and continue exploring Iguanas West, part of the Iguanas hydrothermal vent site. We don't get anything from the InSAS when it's mapping. There's no waterfall or anything. It's just nothing. Afterwards, then we get something, but there's no live view for it. No, we get the data live. It's just it's not visually interesting in any way, shape, or form until it's processed. If you want to know what it looks like processed, if you go to just the cruise page for this cruise, up at the top, there's basically an image from like what InSAS spits out. It's pretty cool, but unfortunately we don't get like a waterfall as it's happening. We're getting the data. It's just I don't have anything to show you of it yet. All right, so the lander is at the surface. So we've broken off from our InSAS run. We're going to go collect the lander.
Good, that was not known, I don't think, was it? Or it was known?
The location was.
The location was known.
It looks like we have a whole bunch of waypoints around it.
Yeah.
There's an index for waypoints, what each number means.
Where is that?
This came from. But in the QGIS, I also have a catalog of all of the events that were in the past.
Is this known and sampled?
It looks like it is, yes. From all the points that we put around it.
Oh, yeah, very nice. Does it have a name?
Aren't we just at Iguanas East?
Oh, the area is Iguanas East. Oh, that's pretty robust.
I mean, it looks like you are belong.
So I'm trying to like log one.
That's pretty awesome.
And I'm also doing
Very nice. So we'll come back here, full workup, so to speak.
I have no idea what's going on. We just switched to these right now.
No, I'll come back.
Somebody call me? The bridge said we were about 100 meters away from getting the.
black smoker just a couple of weeks ago when Roxy and them were here.
Yeah. So,
Yeah.
So that's one that I've been to. Did they give it a name?
I don't know the answer. It's the same one. It's the same black smoker that they saw in 2015. So
it already has a name.
again, I don't know. I just go this way. There's Iguanas East, and it has all black smokers that they just marked.
Okay, so that was just the line and we were not staying there.
I don't have any points from the
Whenever it
most
was
recent
recent, the
cruise
moment
here,
that
just the break.
the moment I come back,
You're too slow.
So in the NAS, there's all the biological observations from all the annotation, and that's what's on
So
the later mass right now.
that one is important.
and the moment I
Do we have
Is
a
it already?
key for these numbers?
Yes, that's the key that I was referring to.
Oh, okay.
So I don't think it was there.
Yep.
Which one got all the observations here in the trial? Go back to
All those numbers are from Roxy's expedition. All those numbers are from Roxy's expedition.
Can you run with that something?
I'm using my hand.
There's Silver.
Texting and flying is frowned upon.
Yeah. Silver is 51, black smoker 52. Oh, so that should be it?
Or we make slender right away.
Roger that. Ooh, I got my life on that.
I'm just
Water. Thank you. Where's this little biology where we're taking this?
Because it just has the
So we're right there. There's a skate. Are we interested in a
Yes,
skate?
what do you see?
It's right down to the
Can
right.
we annotate skate egg, please?
Got two of them.
Oh, yeah. Okay.
Two.
Right
There's
there.
one right there.
Yeah.
And then one around the
Just
corner. I
around the
believe.
corner, yeah.
So that one looks like a
Yeah,
viable
right
one.
there.
Oh, there's
Two more.
3. Are we having to book you somewhere? Is that what we're
They just got the.
up to right now?
We can't really do. We can't really do anything.
Okay, could we just get a Zoom?
I can do something. What do you want to do?
Can we just get a Zoom to see if they're viable or not? Thank you.
What do you look for?
Skate eggs.
Oh, well, if you need, I can make it.
Oh, to look like if they're cracked open. And can we have lasers on, please? So that one looks good. Can we just paint it with the lasers? Yeah, that one's good.
Oh, you want actual on it or?
That's perfect. Can we go look at the other ones now? Looks like there's an old one in the back there, and then that one looks good.
Can you give me a little?
And
There's a
then
couple in there.
there's 2 there. Those look good.
And an
Old
old one.
one there too. Okay. That's good. And then yeah, if you could just peek around the corner.
So there's two possible good ones in there?
Yeah, it
One
looks like
old
there's
one?
2 possible good ones, an old one. Could we also drop a marker here?
Got it.
There it is, there. Could the marker just be called eggs? Oh, you got it already. Can we enter a marker skate eggs, please?
We did.
Ah, that's awesome. Where was I?
You were standing right where you went.
Yeah. And then yeah, if you just paint that one so we can get a perfect. Thank you.
How do they compare to the Canadian eggs?
Smaller, actually. Good question. Those are really small eggs. Perfect.
There's a couple more up there.
Oh yeah. Yeah.
Yeah, there's a whole bunch of biology right here coming up the hill. There might be some density a little bit higher.
So there's another 2, 3, yeah. If there's time, and you could paint the lasers before we move off, that would be great. So I realize that one's tucked away, but maybe the one to the left. We don't know how many they lay at one time. It's pretty interesting. These all look the same color. Yeah, that's
Probably
good for that
because
one.
we established stable DT ready for a year or so.
Just stand by there.
Roger that, just stand by there.
Very cool. Okay.
Oh, it's got a snail on it, see?
Oh yeah. Can we get a zoom on that?
You chomp. I'm sure he's looking at it. That's all it's.
Yeah.
Do you know what all the white stuff is?
Oh, that's just forams pouring down from above.
Okay.
Very cool.
It's just plankton.
Okay. Oh, there's one just above it too.
Yeah.
Even if you just drive over it with the lasers, yeah, like that, perfect. So that one was fireball as well, it looks like.
Do you know how long it takes when you go dark?
They don't all go that dark. Like the Pacific white skate looks like it stays like that color, and they only go really dark after they hatch. And there's another one there. Yeah.
You're gonna paint them all, or just have us go around saying what you got?
At this point, do you have to like move on? There were two right there.
I don't know, there's nobody sitting here, so if you sit
Okay.
down, it's
But like, are we heading somewhere because we're following the ship, or are we just like
We're
killing
not right,
time?
we're not right now, the vessel's got the lander, and
Okay.
we're ready for y'all to tell us pretty good.
We're good? Let's go back to the black smokers.
Okay, you don't wanna investigate this area real
It
quick?
did have.
Yeah, we can do that
Yeah.
too. We
Yeah.
can do a circle around.
So what is the next move? Are we going to start doing biology again, or do we need to do
All
some
of the
rocks?
above. All of the above.
Okay.
Water, rocks,
Because then I
smoke,
would ask for
biology,
Actually,
some
would I?
video surveys. Is that what you're asking for?
No, just I mean, I'm happy to see the eggs here. I would like to do the collections closer, but at least if there's nothing there, then I know where there are some eggs.
We're not that far from the smokers, so I mean.
Yeah.
Like you see my little markers that Carl has right here?
Yeah.
That's where the smokers were, so I mean.
Yeah.
I came up quite a bit from there to where we got to, so I could follow that trail.
Let's
See
do
if we
a
got any more.
let's do a bit of a box.
There's no heat, right?
Basically, swing comes from where you are now
No,
and swings
not right
around
here.
that
But they
that
go
fault
up
there,
like 100 meters.
and then back to the
Another
smokers.
There's another one.
Come around.
Oh yeah, there's some up there.
Swing around.
There's another one down
There's
here.
some-
See this?
okay.
Down here?
Could we track to the left of this?
There's one on the right side.
That one looks very new.
Wow.
Yeah, there they are.
Another one right to the right.
Oh yeah.
Wow. That's a very new skate egg.
Yeah, look at the curlies on it.
Can we paint that one?
Yeah. The
Thank
picture
you.
on it, beside it.
Wow. Can we sorry Mark, I know this is your show, but
No, no,
can
I'm
we,
just here to prevent any chaos. But that's
can we
clearly
zoom
not
around
possible.
this feature that's,
Yeah, that's what we're doing.
just rotate around this
That's what-
oh just the little feature that's in front of us that's only a couple meters
Yeah,
big.
yeah.
Just I don't have any of my paperwork here or anything, but
I'm trying to
Alright,
so we have one there and there. Wow, and here. Very
There's
cool.
another one.
So if we could just kind of like look around this area. What is going on there?
Are they like white? Why are they like?
Maybe it went rotten. They're right in there. I'd love to zoom on that white one. Yeah. Yeah, it's been in there a while. I wonder if that crab could this be giving off heat? Is this a little bit of bacteria in here?
Like the white on it?
Yeah.
Yes, you're right.
Do you think it's giving
Yes,
off heat?
yes. Well,
about
it's
that
giving
diffuse
off
flow?
sulfide if it may not be heat.
Talking about where that bacterial mat is like
Could
that?
see them
Could
now.
we sit down here and
Yes, come here a little bit.
yeah?
Do we already get the lander?
Yes.
Yeah.
Yes. Yeah.
Are the eggs? So
No, the question
Sorry,
is
you wanna
whether.
see the white one?
Yeah, the
No,
white one, and I'm
proceeds
wondering if this is giving off
faster
heat.
when it's more-
Like it's covered in white. Sabrina, at first I was thinking, oh, this is just like a rough structure that's sticking up.
But maybe
And there's
some
eggs
of it-
actually sitting on it, but
Yeah.
maybe there is a bit of heat to it.
Well, the bacteria say that it means there's sulfide coming out, but not that they want that. And there's supulate tubes here, which suggests that there has been venting.
Can you find Charles so that they were so
I mean, they're right down there. Yeah.
John's computer's right there, but
Try to get a different angle, this is not what I know.
that map of the market has targets and Charles.
Just looking at crush right there, that one that is blue.
No.
Doink.
I promise I didn't eat it. I was doing everything.
I mean,
There's
okay.
another white one right there.
White
Oh,
thing
there
down here
is,
looks like
yeah.
Clam down to the left.
Is that a clam or? a-
Maybe we could take a look at that one there.
That's a clam.
No, it has some fur stuff on it. That's another egg.
Okay,
Zoom
no,
into
you're right.
it.
So it is.
Yes, yeah.
Let me,
So white.
We saw some with the white stuff during the Octopus Odyssey cruise as well. I'm not sure if we looked at one.
We collected stuff with fouling on it, and they were rotten, and it was not
Yeah.
a good collection. But maybe we could still take a look.
Did you want to collect some?
Yeah, I'm actually thinking about a collection here. I'm just running through my head because we're going to be going to. I would have liked to have done a temperature measurement in there, but I know we were struggling to.
No,
to
we're not.
sit.
We're done.
I can.
Oh, to sit. We were like bouncing off the rock
Oh,
there.
yeah, I can try to get you there.
Could we zoom in?
Oof, that looks like it's seen better days.
Oh. Yeah. That would not be a good collection.
But look at the bacterial map on it.
Yeah.
So is it being cooked? We got boiled eggs here?
Connor, I remember a couple on the prior expedition.
I'm joking. I really don't.
Okay. I am going to ask for some collections here. And I'm going to think for a second. But I also want collections at the hot places.
You can zip around or try to get the parking spot?
I'm actually.
We need
I
the
don't think
points.
that's going to be a viable one, but
You want a violet one?
yeah, I want a whole bunch of different ones. But
Like the one that just fell down, the gold one?
yeah, I kind of want to get a range of them. I think that now's the time to like establish development. And can we? I'm gonna go grab my binder, but can we measure with one hand and then pick up with the other hand? Could we be holding the probe, and do we have to put the probe back when we fly around?
We don't have to.
Okay.
I would have to, are these going in the
Yeah,
box?
they're in that folder.
Yeah, in case we didn't want to find fireboxes.
We can definitely try, we can hook some with it, because our board
Yeah.
has the finger.
Yeah. Yeah. So the protocol would be to put a thermometer like underneath where it's touching, establish what the temperature is, and then pick it up and put it in the bio box. And we'd be putting all the eggs from here into one single bio box. It would be like one sample, but we would collect a bunch.
And so you want to find an area that has quite a few in the same spot, or?
Well, actually, so basically where we are now, like, you know, I'm not sure what the state of this one is going to be, but I'd like to collect it. So we do it here, and then we get up. And what I would really like to do is to be able to try put the temperature probe into that crack, and where it looked like there was bacteria. Yeah, get that one, pick up the yellow one too, that temperature measure won't make any sense because we just moved it. But we'll have this as like one site. Yeah.
And you may be able to match the temperatures of the particular egg case.
Yeah, given
from
how
the
different.
imagery.
Yeah, given how different they are. Yeah.
So we need to get the high temperature value.
Yeah, and I'm gonna grab my binder while you guys.
You're expecting a temperature measurement of
So
an anomaly here?
So each egg will go with a temperature measurement. So no, I don't actually expect one here, but like for the study, it would be a temperature measurement and egg, and then I do expect some to be anomalous because some are actually sitting on mussel beds.
Cherise, do you want us to note the temperature when we write down the sample on our
Yes,
sheets?
that'd be great.
Okay.
But each collection varies. So it should be in sampling there in each egg case.
Yeah.
Even though if you just in the observation
It's on its way.
temperature and continuing the collection sample, whatever it is.
So there might be more than one temperature range.
There may be more than one sample.
Yeah.
Samples, yeah.
Okay, the blue dots are the dots of interest.
Yes. The blue dot is where ROV is, but I need to make sure it's up to date. Yeah, it's up there, and then we've got it, and then it's like 30,000. Okay. Okay. So Iguanas east.
Yeah, I know where we are, I just want to plan ahead to where we're gonna go. It's swing down.
Yeah,
Well, that's gonna be hard for that one.
Oh, yeah.
Yeah, let's finish this one first. We have bags of time, so we'll look. And we'll look at this one. So we'll do a circle around here and then go this way.
So it is possible to poke through these eggs. So underneath the egg. Yes.
Somewhere.
It'll come out a little bit more, and once you get down, I'll work out a little bit.
So we want to measure the temperature of what it's sitting on. So if you can like nudge underneath it so that.
Can you go and ask us what the numbers back on?
But like I said, it is possible to go through the center of the spot if they're, especially if they're squishy.
Here.
What I want to do is collect this thing here.
So we'll do.
Oh yeah.
We'll do a survey here. We saw all the blast markers right here.
Yeah,
Right there.
that's Iguanas East.
So I figure we'll do it. We'll go around here and then we'll come back and we'll do all the sampling there.
Okay.
Temperature, water, animals, sulfides, anything else? Do you want to do a photo search? I mean, the Nautilus guys did a pretty comprehensive job here already, right?
Yeah, we don't need
Yeah,
to.
so we could just clean that up, go back, maybe look at this structure, and then go and look at all your question is, do you have a preference to go in this direction or towards penguins?
Hold on, one at a time here. Collect rocks, low T, high T, south, push close. Glass
Oh, yeah, so 64. There's some opacive.
So the question is whether you want to go, you want to work the way back. I can't remember what was here.
No, there's some black smokers there.
Oh, okay.
I feel like you're under it now?
Because you've got all that for another dive probably, or a couple days. Okay, so we'll work our way back to there.
Yeah.
You can go to bed or whatever you want to do.
Well, we could.
So we're tucked underneath it?
One back here. So Chris is behind his head.
Well, we could do a
We're like
sample for a
tucked
while.
underneath it?
We have to-
Okay,
we have to
great.
collect SAS
See.
at some point. So that could be like an overnight.
Okay, so
The whole thing.
just hold it there for a moment.
Yeah.
So basically ambient.
We're coming out of the woodwork.
4. Okay. No. Okay.
2.64.
Yeah, it's kind of, that was what ambient was jumping at too. So if we could collect that one.
We could do 10, 12 hours. But I'm thinking maybe we do that overnight.
Okay.
Or we fill up the ROV with
Fill up the
samples.
ROV and that dictates when you come up.
Come up and then go back down and then back and then back to sample. Are you gonna collect?
From this area? Yeah.
Are you gonna fill up?
Well, I'd like to do at least 5 here and then move on. I want to collect around Black Smoker, so I don't want to use like all my collection here. Yeah. But if we're in like collecting and looking at mode, then I'd like to like just be sure that I can use this time. And
Yeah, if
collect
this is
stuff.
what we're doing, then
Yeah.
we do it. There's no reason to.
How do you want us to log this? Like, each one of these eggs will be one biology sample?
I wasn't. I was gonna do like all the eggs that go into one part of the bio box was gonna be one sample.
So this one, for example, if you collect it, we should put it as
I would
five.
not split up samples just by where we're putting them. If you bring multiple, like either it's one big sample of
It's one
8 eggs.
Yeah, that's what I'm saying. It's
Oh, yeah.
one sample of 8 eggs. Yeah.
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 So I shouldn't log this one then? Like, when I say log, I mean writing it here.
Yeah. So you could write it there, and you could say skate egg is bio like whatever 2.
B05.
Okay, and then so write skate egg, and then in brackets we'll end up putting how many are in that sample. So like there could be 8 in that one sample, and then when you log it in there, you can just keep writing the same number again, like sample
Okay, right
5
now we're
again.
on
Yeah.
05.
Yeah.
No, there is bottom line, yes.
Okay.
Nice grab.
Yay.
So it looks like there's something in it still. That's great. I hope it's fireball. The color suggests that it will be.
Just the first one.
I don't have any pictures. Do you have a picture? Scott, can I see this? Oh, thank you. BB2B.
2B?
Yeah, one of the front ones.
Sweet, that works.
Thank you. Description. Around. Oh no. Put it back. No, I'm joking. Don't put it back.
Just double check. That's in there. For a while. Yeah.
But we will potentially be adding more
cascade eggs to sample.
I'll just have like a pint or two or.
Zoom out. One thing that Monica needs is those photomosaic transects, straight lines away from active. The problem with those is it doesn't leave much opportunity.
Can you confirm the sample number? Sorry, I know you told me.
B05. And where is it going? PB what?
300 meters.
BB2B.
This is an active egg. One's out there,
Yeah.
We end up dropping it.
The sample went into 2B.
It went in or-
but
Sorry,
the
I was-
bacteria fell off into 2A.
What fills into
The bacteria fell off into 2A.
It's like a sheet.
Are you ready for the next request? Could we go back to where we were looking into the crack, where we could see a few eggs and see if we could set up there for checking the temperature inside?
So, yeah, like this is let's fill it up. So we're gonna fill it PRV. Go back. So, I don't really want to tape. So I don't want to get some sleep.
We don't have to.
Could just, but then people want to get going on their samples.
Oh. I know that we knocked it, but maybe we could just grab that egg. Sorry, I know I'm changing my mind. Yeah.
The flight characteristics.
Okay, so there is a very bright, it's a very bright egg down that way.
It's that one right there.
So we won't go temperature measurement on it because we knocked it and it fell down there, but if we could just collect it.
Yeah, the decision to SAS now or later comes probably down to what's most time because they're ready for sleep. So it'll take us several hours to collect eggs and then do transects, collect sulfide. So that'll be midnight at least. So that might be a good time to nap because when I jump out of bed, come up in the morning when we get their samples off and we'll go back down and do more.
Sample is that all night and then
Yes.
How do we preserve them? We're gonna open them up and take out the embryos and do a whole bunch of measurements and do genetics from them and then, yeah, on the boat, yeah. So there'll be a lot of processing to do tonight, yeah. Yeah, no temperature measurement because we knocked it off the area above. So we just do a collection.
I don't know what.
Wait,
Sorry.
no. What's it, Chucky? Yeah. Can we turn the lasers, and paint it once just before we bring it in? I hope painting it makes sense. Means like put the lasers right on it.
They're the ones we're trying to hit.
Perfect. Thank you.
You're down in here. And
Okay, great.
there's no-
Thank
you're
you.
flying
Later
here.
for sample.
You're hitting them that way. We're over here. You're in, I think.
Yeah, I'll have a lot to process when this sub comes up.
Because if I ask you to send me what's just to let me know they're coming out or whatever. And
Yeah.
and they have an hour review.
Yeah, I've been doing that for people.
Okay.
Yeah.
Please, because I do that.
Absolutely. Yeah, 100%. Yeah.
trees. Can you tell us about the eggs?
Yes, for safety.
What happens when they hatch? Well, you've never seen that happen, right?
No, we haven't seen that happen. Okay, so these are the eggs of the Pacific white skate, and it lays its eggs only what we know of in 2 places in the world. One is right here where we're diving, and then the other one is in off Canada in the Pacific, and they lay these very large eggs. They're golden in color. They turn a little bit brown when they've been down here for a while, but they have to sit down here for like 4.5 years, incubating to turn into viable skates that then get to swim away. And what we believe they're doing is taking advantage of the heat that the hydrothermal vents are putting out to create stronger individuals that maybe don't have to incubate as long. Yeah. I have no idea what that. I think they're talking about the flying crab there. I don't know if anyone else could hear that, but there are a lot of cutesy noises being made in the background.
Do we know how long the skates live?
Actually, we don't. I'm hopeful that there's colleagues from around the world that are looking at the skates that they've caught to make a growth curve to figure it out. The estimate is decades, but we actually only have 7 specimens in the world of this skate.
And wow, do you think that they lay? How many eggs do they lay? And the sac is already this size when they do?
Yeah, so this is actually a lot smaller than the ones I was seeing in the summer. So we're looking at like a 10 centimeter, maybe a 15 centimeter egg here. And the ones that we were pulling out in the summer were like 30 centimeters, 45 centimeters. I was looking at a paper yesterday that the largest embryo to come out of these eggs was actually 26 centimeters in size. So that's like a dinner plate skate that can come out of the species eggs. So these are very impressive eggs though. These are huge by any means. I'd say the largest of like the skate family, but for within that species, these ones that we're looking at right now are actually quite small.
They would lay 1 egg.
One egg, yeah, but they might lay 1 egg at a time and then lay like 4, maybe 8, like over a duration. Yeah, but the neat thing is there's only 1 egg within this egg case, and some skates you get like multiple in one, but these skates seem to really put a lot into their individual like young. You got 1 big egg with 1 big embryo in it, and again, they are probably choosing these hydrothermal vent sites to speed up that incubation and to make for like the strongest individuals. So they're really putting a lot into each skate's egg that they put out there. Yeah.
Are you looking for sampling eggs of different stages of growth?
Yes. Yeah. Yeah, so I'm going to use the color of the eggs as a cue for stage. This one seems a little slippery. No, yeah, totally. In Canada, we're referring to them as bananas because of their color and how slippery they are to pick up. Would it be helpful to grab the horns with the other manipulator?
Try not to get too aggressive with it, but
Yeah, you can kind of see that it's like, so the young is in here, and then this part and this part here is actually quite good for grabbing, and it's really the center part, the center of the ravioli, if you will, that is like the squishy bits.
Does the size of the adult change depending on the size of the egg?
We don't know that. 7 specimens in all the world. That's it. And a paper was only published in 2022 that claimed to be the first description. So we don't know much about this animal that lives at 3 kilometers down. It hasn't got caught by our nets much. We haven't retrieved many. So there's a lot that remains a mystery about this skate.
got 2 more questions.
Okay, but if you got a quick one, because we're probably gonna ask for another move now. Although J-Rod knows that I'm hoping to
The one
fly
that was
up.
hatched.
Yeah, the one that was hatched. Thank you. Okay, so we got our second one. So now if we could go up and perch, and then we'll try sample in that crack. Yeah. Yeah, so rolled. Sorry, I feel like I'm all over the show with answers and noises and yeah. Yeah.
So I'll probably go in.
Yeah.
And if you could please send me a message when there is like half an hour.
100% will do. Promise. Yeah.
You have me on the group.
Yeah.
It's okay. So, no, let's try. Anything else? Maybe you can change the
Yeah, that's the area. Look how it's sitting right up
symbol.
there. It's
Remember?
amazing.
Yeah.
Yeah.
Is that the white one was?
Yeah, it was. I thought it was in here, but now that we're here, I'm like, how on earth did we see it? Oh yeah, you can see it there on your screen.
Just want to make sure we don't retrace it. Remember that it's not accurate. That's what
What
Not a
aspect of
list.
it?
Yeah.
What aspect of it is not correct?
So I loaded the Hercules track. That's the correct one, and the KML doesn't track it.
Is this the KML or the Hercules track?
It was a KML, but it makes sense where it's going. So
Yep, that's fine.
it might not be accurate, but not super far off.
Yep.
Yeah.
So the
So
egg that
could we paint the ones with the lasers that we can see right now before we start collecting? Oh wow, there's so many down there. So
Just gonna grab all my stuff.
no problem. Sorry.
No, it's fine.
It's just Canadian of me, I have to say sorry.
Yeah. What are you
Yeah.
apologizing for?
I don't know.
Like a UFO.
Like a waffle.
Like a UFO.
Okay, that's great. Jared, do you mind if we look at more of the eggs that were in the crack before we go collect this one? So we painted this one and this one. Could we look at this one too with the lasers?
Trees. On that one, there might be a snail, so you're not zooming. There's a white
Which
ball.
one?
The one you're zooming right now. There's a white ball.
That one up top there?
Yeah.
No, not that one, but if you look at this.
This one doesn't look like it has anything in it.
That's it. Yes.
Oh, yeah, it does.
Yeah, this probably
Yeah,
could
this
be a
one
first
looks
step.
different, eh?
Yeah, that's
Yeah.
another one. That could be a first step.
Is that what you wanted to see, John?
Yeah.
Yeah. Okay.
So that could be a first edit. The one that only leaves.
There's a lot down here.
That guy.
Yeah.
And all of those in there.
Okay. And all at different stages. This is really cool. Okay, so if we can go back to the white one that was to the left, and are you able to take your temperature probe and go into that crevice? And then could I have, or am I missing where the temperature is for up here? Oops. Is there a temperature?
Yeah. You put the height in purple?
Yep.
Try to grab it with showing the top.
Is that what he said? It's supposed to be transept. Okay, really, yeah.
Good country. You kind of broaden from our time early or something?
Right, so when we're done with this, we're gonna head back to all those smokers we just saw.
Did you look at the boiling? I was.
So
No, this is the
I'm
total
hoping if we could just get into like this area here, just anywhere and see, just kind of looking for is there any higher temp coming out of there.
Anyway, so we can do the 300 meter, what was it, towards sites from the site that we're near. These are the chimneys here. We'll just up here now and do some, come back and do all of them.
You think that one is older than the others?
I don't know, the color is kind of bright, but it is covered in bacteria, it looks like. It could be like where it is and it's just gonna be covered. The color suggests it's like not that old.
We don't know.
Really?
Not as, so we collected one that was bright yellow and that would have been the freshest one we've seen.
I thought it looked pretty brown on the ends.
I don't know if you noticed some of them, they're like almost black in color. Yeah, but you get viable ones. Yeah. Ooh. Okay. 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 So if we could, collect it now, that would be wonderful. Thank you.
Mine for that hole and your jaw. So we just set that down.
It's just 25 seconds and everybody has their own pace.
The one that was hatched. It looked like that there was a hole. It didn't look like it was torn. It looked like it had melted off.
I don't recall that. But if the top is broken open, then the little guy escapes. There's like a weak spot in it that lets you get out. If the soft bit, the belly bit, has a hole in it, then it was pretty. Yes.
I'm taking the temperature for each one of them. How are you going to be able to call them up, or is it even necessary?
Yeah, so I'm going to be taking lots of photos of them up here, and I think that I'm going to be.
Yeah.
Yeah.
Yes. I'm taking a lot of photos of them.
Thank you. Right now I have nothing being over ambient.
Yeah.
Yeah,
278 was the highest.
Yeah, I'm having ambient go between 2.49 and
So here I'm
2.64.
logging ambient temperature and the max
Okay.
temperature of the probe.
I can try to bring that forward.
Andrew's back.
Alright, you're clear to shoulder down. You also gotta do a mechanic. Rotate counterclockwise, so go with that angle. If you pitch down a little bit, come up, down, might help you drop. There you go. And then we go from there, then you go going at that angle.
Sorry. So we're doing that now.
Finish off the correct.
somebody else.
No, I started researching. Erin was here for longer and then she went on to eat.
I'm happy to write notes and stuff.
That's
But if
helpful,
you want
yeah.
somebody else to,
It's government issue laptop, so I need to have my kid card to be able to use it. Yeah, it has a chip, yeah, and that's how they, so then like, I plug it in and then I have to put like a passcode and then it's me and I can access it.
Hey.
yes. But you can take it if you want. So what we've been doing is all these skates.
Some paint on it for some reason.
You just scratch it that times 2 and then times 3. So it's all in the same box, same center. Yes.
Well done.
How many more are you gonna listen to?
I was hoping for 2, but they're not really lying down like I thought they would. Ooh.
We're gonna easily see what this is. Might have to.
Reposition. Okay. Okay. So I don't think any more right here. What I'd like to do is circle this geological feature and see if there's anything that looks like obvious venting, and then sit down and collect another 2 before we leave here. But I just want to see if there is any obvious venting.
These rocks are all like bright red orange inside.
So I might be just gonna think out loud, but I might be interested in that one there, depending on what we see when we fly around. So yeah, just around this area here. just to see, and if we're close enough to still like see the skate eggs, but we won't paint every single one. We'll just look. Okay. That looks like another one right down there. Can we just zoom in on it? Just to see. Okay. That's good. Ooh, you're gonna paint it. You got it pretty close. Okay, that's perfect. Thank you. Yeah. Okay. Yeah, see, you can see that one's broken open. That one didn't fare well. This one up there. So if we keep going around to the right. Thank you. Pretty interesting. There's one up there.
Is that Matt?
Yes. Yeah.
Possible lifting like that?
Karina, do you think if we sat down that's enough, Matt, to suggest heat, or just trying to find out if there's a heat source here, so I thought I'd fly around a little bit to see if there was anything obvious. No.
Oh, here's some up here.
Okay.
See right there, top left of my screen?
Top left. Oh yeah.
Oh, which one? The main
Yeah,
thing?
yeah, yeah.
Yeah, that would be worth
I don't know.
investigating.
Yeah.
Shimmer, but.
Well, it still doesn't have to, I mean, you still get a little bit of a heat signal without shimmer.
That's really neat. Okay. Yeah, if we could get a temperature measurement in there. Thank you.
So.
Starboard wind is coming up. I'm playing quickly.
I wonder if the females cue in on sulfide.
Yeah.
In the water. Pull right in, that's that hydrothermal, it was way out here.
It definitely looks like this area has been used a lot over
Yes.
time.
Yes.
Yeah. Like lots of different stages of decay.
Anywhere in particular, or?
Right in front, Manson, yeah. Yeah.
Gonna bring the foreshaddow over, and we'll,
It's the big conundrum in metaphors, because technically you cannot carry the metals and the sulfur in the same fluid, because the metals and the sulfur will combine, so they have to make sulfur.
You want like poking the mass or just in the water?
Solutions.
Uh,
Fluid
there.
very acidic.
That's
Say
good.
as far as you could get into the crack.
As far as you can get into the crack?
I would suggest
Okay.
that.
Case you
Yeah.
could like also
I
right.
mean, just down to the right where you can see what you're doing there.
Oh, right down here.
Yeah.
Yeah. So up against that rock didn't give us anything but ambient there.
Could be more shallow circulation, could be cooling fluids circulating deep.
5 seconds.
There's a lot more sulfur.
Okay, that's good to know.
So you can precipitate over. Still have plenty of sulfur.
Gotta wait 5 seconds.
I got this. I have real time up here.
Oh. How come you got the good stuff?
Okay.
Okay.
That's disappointing.
Maybe while we're holding it out, we just, I don't know, see if we can reach over to the right a little bit more and see if there's another crevice. Oh, hey. Anyone else see that?
I saw that.
Yeah.
A big anomaly. We're all excited.
A little jump.
Yes.
A lot of sassy people in here. Okay.
I think that was
Okay.
Just friction.
that was the question.
Or the ROV turning to the right.
Yeah,
You know this
that
one?
seems to be right.
Everyone's a critic. That looks like a good hole right there.
With that weird screen on this?
Where the weirdy thing is, yeah. Yeah, Kira, yes, that's, because the thing we saw before was too long.
Oh.
78 again. More friction.
Stop it. We're excited.
Now you're trying to get a whole bunch of friction.
Yeah.
Oh, no.
It's gonna start a little fire.
Okay. Well, whatever it is, it's not very much. So
Bring it up.
yeah, let's
We
bring
don't
it
want to
up.
break that.
No.
Also, anyway, it's not going to be a anomaly with end zones, so it's irrelevant effectively.
Well, so I wanted to get what the hottest vent was around it. Yeah.
But it's definitely not that, because
Okay, so if we could collect another 2, maybe I'll just.
White, yellow.
Not bright yellow. There was one that was sitting on the top that I like the look
Is that
of.
really dark one?
Not this one
No,
here.
the one that was sitting at the top of the rock?
Yeah, yeah.
All right. While we're repositioning, I'm going to restart the streams. So if you're watching on YouTube and Facebook, this stream's going to end. Part 2 will follow. It'll just be a separate stream. If you're on YouTube, it might move you over automatically. Otherwise, keep your eyes peeled. We'll see you on part 2.